due to funding. Anyway, I thought it was really important to come and engage with all of you in terms of, and much as I said here, the why, the what and the how of biocontrol in South Africa, I'm going to focus on weeds of the Western Cape rather than countrywide. I don't know if you guys can see the screen. Fine. Okay. I also think it's really important, I wasn't too sure of my target audience today, so I'm pretty much going to start from scratch in terms of the whys, and a lot of you may be very familiar. A lot of you might have attended training courses that I've done in the past or talks that I've given, so apologies if you're hearing it all again. But I suppose, first of all, why do we have invasive species in the first place? And I think all of you guys sitting here today know this. I mean, plants were introduced into South Africa way back in the 1800s and earlier, intentionally, many of them, because they were brought in as ornamentals, they were brought in for shade, all sorts of different uses, and they were later widely planted because of their uses. You know, black wattle was a great firewood. It was wonderful for tannin production. Black wood, excellent for furniture. But many weeds that we have in this country were also brought in unintentionally. And they really have very little use at all, but they've become extremely problematic. The one thing that all invasive plants share in common is that they have all been introduced into South Africa or wherever they're introduced in the world without their suite of what we refer to as natural enemies. And those natural enemies are generally the things that keep things suppressed in their country of origin. And it can be a range of insects, it can be pathogens, it can be mites, nematodes, even larger herbivores that can just keep things suppressed. And essentially now what happens is that these alien weeds, you end up, like on the top left-hand corner there, very dense stands, very thin, spindly stems, which are useless. You can't even use it for firewood. They are massive fire hazards. They threaten our water and limited water resources. And they also compete with biodiversity. So there's nothing really good about them. And, you know, if one starts looking at cost-benefit, a lot of people have done cost-benefit analyses of alien plants. And generally speaking, they're more of a cost to the country than they are a benefit. Okay, now obviously again, you guys are very aware of the various management options available for invasive plants. There's obviously the mechanical line, there's the chemical line, and then biological control, which is what I'm going to focus on today. Okay, so what is biocontrol? And I think a lot of people have a vague understanding. And this is really one aspect. There are different forms of biocontrol, but the one aspect is when you introduce your biocontrol agent into a region that is not part of its native range, and it is introduced in order to suppress a selected target weed. Generally speaking, the biocontrol agent and the target weed come from the same area. It doesn't always work like that, but generally speaking, I think for purposes today, let's look at it that way. So, for example, Australian acacias, they were introduced from Australia. So their biocontrol agents have been sourced and introduced from Australia. So it's a fairly simple process, but it is a very scientific process. It's not just a case of grabbing an insect or grabbing a pathogen, bringing it out and chucking it out into the country. So if we look at, and this again, it's a diagrammatic representation and it's theoretical, but what happens when you first introduce a plant or an invasive, particularly an invasive plant? So it starts off down, my mouse is going to work, but down here at 0.0, and then it starts building up. So the weed, the populations of the weed build up, and they go above what they call an injury level, and then they kind of plateau out. They reach their point where they've covered all the area that they can cover, but they don't stay stagnant at that height, at that sort of density or producing that amount of seed. You get these fluctuations, and that can be because of fires, because of clearing, because of flooding, all sorts of things. So these are normal processes. But what you have is this quite high equilibrium level. before any control is introduced. Okay, and that's obviously not what you want, because that's what's making the plants problematic. So the idea is you introduce your control agent, and unfortunately, as I was chatting to Glenn last night, unfortunately, biocontrol is often introduced rather late in the system. You know, people suddenly realize that they've got this problem, that the weed has really covered all its extent and it's become particularly damaging. People have had this sort of issue with, oh, you know, they can't, chemical control's not working, mechanical control is difficult. So people say, oh, you know, let's get biocontrol. And that's not really the way we should be. We shouldn't be seen as a rescue service, because actually biocontrol works far better if you can nip things in the bud. So if you have a plant that's been introduced, I mean, we often say, introduce it with its natural enemy from the outset, rather than waiting until it's reached this level where the horse has already bolted. And then weeds become extremely difficult to control. But the whole idea behind this is that once you've introduced the biocontrol agent, either the populations of the weed will diminish, like you see there, and then again, you reach an equilibrium. So you're not going to get eradication. This is not biological eradication. It's biological control, or I prefer in a way to call it biological management or biological assistance. It should never be seen as a silver bullet. I mean, a lot of people, as I say, people expect, the expectations of biocontrol in the past have just been extremely high. And I think part of that stems from many of the really big success stories. So there have been big biocontrol successes with control of cactus weeds, control of red water fern, azolla, where plants have been pretty much knocked back and killed, and populations have really crashed, died out, and that's wonderful. But not all biocontrol works in that way. And actually something I chatted to Glenn about this morning, I think we need to have a slight shift in perception about what biocontrol can achieve. And if you're wanting eradication of a weed, I think in many cases biocontrol isn't for you. But if you're wanting assistance, if you want to buy some time, if you want to slow things down, then wonderful. Biocontrol certainly is the way to go. And again, once you've reached this level, you get what they call typical predator-prey. You know, the weed is still there, but it, you know, some years lower, some years higher, might produce less seed some year, but you're at a much lower equilibrium level. Okay, so essentially, as I said, it lowers the weed's reproductive or competitive potential. In some cases, yes, it can kill plants, but it doesn't eradicate the target weed. I don't know of a single biocontrol agent that has completely eradicated the target weed. Okay, so if we look at the scenario within South Africa, so who deals in biocontrol? Who is responsible for biocontrol? So the main role players are the Plant Protection Research Institute. I think they are now called Plant Health and Protection. They're part of the Agricultural Research Council, and they have units up in Pretoria, Sidara in KZN, and at Vredenburg Farm in Stellenbosch. And they are responsible for importation, testing and release of agents, and doing a little bit of post-release evaluation. It's obviously moved more towards post-release evaluation now because of the funding situation. There are no new agents, or very few new agents being brought into South Africa at the moment. Then they have the universities that are involved. And initially, our role at universities was purely in a post-release evaluation. We were not dealing with the importation, screening, the host testing. That was all done where they had quarantine facilities. So we need to have government-approved quarantine facilities to do all the testing. But in probably the last 10 years, Rhodes University have a quarantine facility, Wits University have a small quarantine facility. So there's been a little bit of a shift, and universities are also making use of, I've been sort of semi-seconded out to Stellenbosch to Plant Protection Research Institute. So I've done quite a lot of the testing for acacia insects over the years using the Stellenbosch quarantine facility. And lastly, the sort of grey area that I put in grey there is our our funding source, which always was, originally was Department of Environmental Affairs back in the day through the Working for Water program. Now has become, they change their name every so many years, but it's now Department of Forestry, Fisheries and Environment. And yeah, for I guess around 20 years, we were extremely lucky. We were incredibly well funded by government, and it allowed South Africa to really achieve wonderful things. I mean, on a worldwide scale, South Africa were right up there in terms of biocontrol in the world. A lot of people looked up to projects that we were doing. There was a lot of piggybacking on projects that we were doing. And unfortunately, I think it's now, in a way, maybe they see, oh, well, you know, you've got the biocontrol agents, we've got more important things to fund. You know, I think people are less interested in funding projects, which, as Aileen mentioned earlier, it's unfortunate, because I think what will happen when there isn't that continued, or there's not the continuation in biocontrol releases going on, bringing in and doing research on new agents, something suffers. And invariably those weeds just get a handle on things and start taking over again. Okay, so if we look at the steps in a biocontrol program, so firstly, we might be told, okay, we have a problem, or we might know that there are these priority weeds. And generally the first step is to look at the literature, and you find out what is known about that particular plant and its control elsewhere in the world. Has anybody worked on it? What can we learn? Then generally, as a next step, you then go and investigate the natural enemies of the plant in its native range. And it might be, so we have a lot of weeds, particularly in this area, that are Australian in origin. There's quite a lot of South American weeds, and we also have some Mediterranean weeds. So the thing to do there is you go over. I mean, I've probably visited Australia about 20 times in my career. It is quite a lengthy process because one has to get permission to go and collect in countries abroad. You have to sort out all the correct permits for collecting, permits for exporting, permits for importing, just to do the initial work, not even to actually release a biocontrol agent. And you can't actually see that because I'm screen sharing here, but what it's essentially saying is that there's also a system called piggybacking on other countries' knowledge. So, for example, I know Australia's done a lot of work on echium. They've got biocontrol agents on echium, and reasonably successful biocontrol agents there. So rather than us starting from scratch and going off to the Mediterranean to look for agents, that part of the project's already been done. We know that there are successful agents, so we could approach Australia and say, right, can we get agents from you? Instead of going this sort of long route. And yeah, it may mean that there's a small amount of host specificity testing that needs to take place, but it's much reduced. So it really sort of cuts back on funding. So one of the things that's really important too is, where possible, when you're doing surveys in Australia, in South America, wherever it might be, try and look at the host specificity of those potential agents in the country of origin. Very often you get when you do these surveys, you get a really gut feel for a particular agent. You can see, well, it might be eating acacia seeds, but it's also eating things in the vicinity. So it's probably a generalist. So those sort of things you want to avoid. You want to do as much homework as you can in the country of origin. And where possible, it's best to try and do some of those studies. Okay, then once you've actually targeted a few, it might be one, it might be 4 or 5 agents that show promise, then what we need to do is apply for permission from plant health for a permit to actually import those agents into a quarantine facility. So it's, again, not importing it to release it, but it's to import it to do the testing. And that can be quite a process. I mean, in the past, I've waited up to 6 months for a permit to come in. It's much faster now. It seems like the process has been streamlined. It usually takes 2 to 3 months to get a permit, and then you're good to go. And then you go and you collect your agents and you import them, ready for the testing procedure. Okay, so those agents are then put into a quarantine facility, and as I mentioned earlier, the main quarantine facilities are in Pretoria, Sadara, Natal, and Stellenbosch. Stellenbosch has the only quarantine facility that is suitable for pathogens. It's obviously a much more stringent, because you're dealing with tiny fungal spores. It's quite a process going into quarantine. When you have insects, which are obviously a little bit bigger, it's obviously stringent still, but we don't have quite the same processes. The pathologists have to go through a shower system before, you know, particularly when they're coming out of quarantine. But essentially, the quarantine facility is a sealed off area. So you walk through. For example, to get into our quarantine facility, you have to walk through a first little room and then a dark room. And then you go into a room where you suit up into sort of hazmat type suits. And then you get into the quarantine labs. There's also negative pressures are quite useful in terms of keeping insects out. They have all sorts of special filtration programs or processes. They are expensive facilities to build, and that's why there are so few of them. They're also incredibly expensive facilities to maintain. And unfortunately, many of our facilities are sorely in need of maintenance. Okay, so then what we do is we do a screening, an initial screening, just to make sure that we can get rid of any parasitoids, any diseases, anything like that, before we actually start trying to work on our agents. And then we need to check the identity and study the biology of those agents. And we carry out what we call host specificity testing. And that specificity testing has changed quite a lot over the years. In the past, there used to be long lists of plants that needed to be tested. You know, and invariably people are worried about commercial species. Grape farmers always worried, the wheat farmers, the potato farmers, etc. But now what we tend to do is we work very largely on plant phylogeny, and we look at the very closely related species. Because in the past, I think we were probably wasting a huge amount of time and effort testing agents that we didn't need to test them against grapes or. So those test lists have been shortened greatly because of the testing procedures. But it's actually perhaps a more accurate science, because we are now testing the very closely related species, and there is I think it's unlikely. If something is not going to go onto a closely related species, it's very unlikely to jump onto something that is not related at all. And this is really all just to ensure the safety of the agents that we're testing. And that process usually takes a good number of years. And it means re-importation of the agents, because it's often difficult to keep colonies, particularly when you're working on agents that are for perennial trees. We can't keep them alive in quarantine. You can't keep big trees in quarantine, and it's almost impossible to keep insects going on potted plants. So it does mean re-importation of those insects. So the initial part of a biocontrol project is expensive, because there's that international collecting, there's that work in the quarantine facility. But once we know that a biocontrol agent is safe for release, we have to write a release application, or a risk assessment, it's now referred to, and that then gets sent to mentioned it here. So it then goes under review. And there are a panel now of people who review release applications. And it may be approved immediately, or there may be some questions which come back to the researcher. It may be that a little bit more work might get done. If it is a plant that has a conflict of interest, it's also important that people who maybe are on the other end of the conflict have a look at these applications, so they can also voice their disapproval or whatever the case may be. And then the last step is obviously when permission for release is actually given. As I say, or there might be additional tests that are required. And then the release happens.
Are there any, there have been situations globally or something where it's gone wrong, where actually there has been a negative, sometimes you read papers where they talk about there are risks. There have been situations in South Africa where it has been, there have been.
So South Africa actually has an incredibly good track record. We now, I mean, we celebrated 100 years of biocontrol in 2018, somewhere around there. It's a while back now. And we haven't had a single problem where things have gone horribly wrong. And I think part of it is that as a group, we've been a very cohesive group. We always used to meet annually and have a biocontrol workshop. Any new agents, before they even got to the point of being, you know, the application for it would be discussed amongst the group, and even amongst the group, if there were any concerns or, you know, if people were wary, you know, often we'd say, right, let's shelve it. I mean, I've worked on agents that my gut feeling is they are host specific, but they're agents that have this really long diapause, so which means they go into like this rest, and that rest can be unpredictable. So it makes it very, very difficult to test agents like that against alternate hosts or potential alternate hosts, because they just never awake when you want them to be. They're in this diapause. Even though I'm certain. So agents like that will say, okay, let's just shelve it. We're not even going to go through the application process. And I think that's possibly why we do have the success. I mean, I think if you look also on a worldwide scale, if you look at biocontrol and the number of agents, a bit like Shark Attack, you know, it's people get excited about the few incidents that do happen. And yes, there have been, obviously there have been incidents, but it's small. And yes, people say, oh, well, you know, you're playing with fire, but it is, and I think that's why I wanted to show you. It is a scientific process. It's not just a case of smash and grab and chuck out, and it's a very lengthy process. I mean, I worked for one agent for, it must be about 6 years before finally getting permission to release it. And one of the reasons was it had just a little bit of spillover in Australia onto black wattle. And that spillover really did, I mean, the black wattle plants would get these tiny little galls, but it really didn't do much to the vigor of the plant at all. But it wasn't, funny enough, it wasn't the wattle industry here that were worried. It was the group of biocontrollers that said, oh, I'd be wary of what the wattle industry are going to think about this. And so it was only along the line when we were able to say to the wattle industry that, look, this is the whole situation. They said, well, they're happy to approve this agent. So anyway, yeah, so biocontrol agents can come in a variety of different shapes and forms: beetles, moths, mites, worms, all sorts. It's not a particular group of insect, so they're from different insect orders. And they also can target different areas of the plant. They can be leaf feeders, flower feeders, stem borers, seed feeders. So we've got a very, very wide range. And obviously, depending on the type of insect, they can be more or less damaging. And sometimes we need more than one agent on a particular plant. It's not you release an agent and you find, okay, well, it's sort of setting the plant back a little bit, but it needs something else to help it along. Okay, and then not all biocontrol programs are successful. Sometimes we go through the whole process of testing and we release a biocontrol agent and they just don't establish. And there can be a number of reasons why biocontrol agents don't establish. Sometimes there can be climatic incompatibility. There can be host plant incompatibility, particularly when you're dealing with agents that, or sorry, target plants that very classically have hybrids. So Lantana Camara is a real classic example of that, where you've got a plant that has all these various sort of types, and it comes from different regions. So you need to try and target, well, where do our specific biotypes that we have here in South Africa, where do they come from? Are they from Jamaica or are they from somewhere else in the Caribbean? Because you need to be very specific at where you target the insect from, to make sure that it's going to be compatible with its host plant. Sometimes other control methods have put pay to success of biocontrol agents. This agent that took me all those years to get permission to release, I had put it into a nursery site on Fredenberg Farm where I do some research, and it established there, and I was really excited about it, and it was the only site where I got establishment. A veld fire came through and it destroyed the site. And then it took me another few years before I could actually get the agent established. I had importations of the agent and I just couldn't get it established. So these things can happen. Thankfully now the agent is established. Then you can have things like predation and parasitism. You introduce a new agent, obviously it comes in without its parasites and predators, but it can pick up those things in this country. And it can pay to the success of a biocontrol agent. Okay, this is really just to show you, biocontrol in South Africa really started back in the 1970s, late 1960s, and it was quite classic it was mostly white men, firstly, out in the field in their suits and their ties, and in quarantine, in their suits and their ties, and nowadays things have changed. So as I said earlier, we generally wear a quarantine suit, which is to keep the insects, you've got to have something over your hair so you're not getting insects in your hair that you're walking out of the quarantine facility with. So it's just made the process much safer. And obviously field work is a lot less casual than it used to be back in the day. So I think that is the end of the first section, which is obviously why we do biocontrol. The next part that I'm going to get to is the whole sort of the nitty-gritty of biocontrol and the biocontrol agents. I don't know if anyone wants to stretch their legs. If anyone has any questions before I move on.
How does everyone feel? Keep going?
You're happy to keep going? Okay. Cool. With this next bit, please, I think it's because obviously you're here to learn about the biocontrol agents, and this is really talking about the biocontrol agents themselves. So if you have any questions whilst we're going along, please stop me or ask questions. Ask questions. Okay, so what I'm going to do is I'm going to deal with the weeds, and I'll deal with the weeds separately and their biocontrol, each of their biocontrol agents. So the biocontrol program against Hackia sericea is probably one of the longest running biocontrol programs in South Africa. And so this was initiated in the 1960s. And it was the first in the Fynbos to be initiated. It was also a world first for using a seed feeder for a perennial weed. It hadn't been done anywhere else in the world. And I think there was quite a lot of people felt, well, what are you really achieving using a seed feeder? And to this day, there are divided interests in terms of whether seed feeders do any good in terms of biocontrol. I'm very for them, but a lot of people just say, well, what are you really achieving with them? But funny enough, subsequent to South Africa using seed feeders, it has been adopted in many other countries of the world now.
So can you explain a little bit more why people were sort of saying it doesn't really?
Well, I think the problem is, and one understands it. So even if you've got a plant, and many of our invasive plants, one of the reasons they are so invasive is that they produce a lot of seed. And in many cases, it's very long-lived seed. So even if you're knocking out 80–90% of the seed on the canopy, there's still that 10% which is available. But it's that whole thing of, again, as I say, it's that perception of, well, what are you trying to achieve? If you're not, really you're trying to achieve a slowing in spread of the weed, less seed being introduced into the soil to accumulate there. So it's sort of shifting the mindset rather than saying, oh, well, they're not doing anything. Well, it's wrong to say they're not doing anything. They are doing something, but they're not killing the plant. So I think that's maybe why there was this. They're saying, well, there's still so much seed being produced. Plants are not seed limited. That's my opinion. Probably put it away. Quite a lot of the biocontrol agents that have been in.
Are there any cases that you have witnessed that when you put a biocontrol agent and it jumps on the indigenous plants?
In South Africa?
Yes.
No.
They have specific plants.
They are, so that's part of that whole testing procedure. So as I say, generally one will test closely related plants. And it is, as I say, we haven't been aware in the 100 years of biocontrol in South Africa of a biocontrol being introduced from another country that's jumped onto a native plant in South Africa. That we haven't had that. Okay, so the biocontrol agents that we have introduced against Hackia sericea also will go on to Hackia gibosa. They're very, very similar species to look at, and I think a lot of people have problems in identifying the two species. It's quite difficult when if you're in the field and you only see one, you don't really have that comparison. But generally speaking, Hackia gibosa has got a longer needle. I mean, the leaf is in a needle, and it's more pubescent. It's more hairy than Hackia sericea. Hackia sericea tends to be sharp and smooth on the whole. So that would be my. Hackia sericea is also much more widespread. So it's the one that is more commonly seen in the field, but certainly there's plenty of Hackia gibosa around. Okay, so there are quite a suite of agents that have been released against Hackia over the years. Erytena consputa, and don't worry too much about the scientific names. It's a little fruit-feeding weevil. Was released in 1970, and what happens here is the female weevils lay their eggs onto the leaf. I call it a leaf, but it's actually that little spike. So they lay their eggs onto the leaf tips or the buds. The larvae, so I don't know if all of you are familiar with the life cycle of an insect. So you know what a larva is, that hatches out from the egg. The larvae actually pull down from the leaf or from the bud, and they burrow into the young green fruit. So before, I think you're all familiar with those big knobbly Hackia fruits. So it's before it actually gets to that big stage. It's still flat and it's still nice and green and succulent. The larvae burrow down, they burrow into the fruit, and they actually feed on the seeds within that fruit, and they stop the seed from developing, or they stop that fruit from developing any further. You then find that those fully developed larvae will leave the fruit and they drop down to the soil, and they pupate in the soil. And that's generally, so as I say here, generally females will lay their eggs in towards the end of winter, July to September. The larvae then drop to the ground, pupate in about October, and then adults, you can find adults pretty much throughout the year. If you go and shake a Hackia bush, you will find adults, but they tend to be most abundant during the late winter, and that's because that's when they're starting to sort of get ready to come out and lay their eggs. And okay, again, that's sort of blocking the thing, but the larvae can destroy up to 80% of the green developing fruits. So what to look out for in the field to confirm whether or not you have Erythema present? So what you would look out for are these classic, what we call husks. So at this stage, they're green. Now, obviously, all the green fruit on a Hackia plant will look like that. But as they start maturing, those just dry out and it just becomes a little brown husk. And they are pretty common, they're pretty widespread. It was always felt in the past that this agent needed to be redistributed. Particularly, Hackia is one of those very fire-prone species up in the mountains, and the feeling was that it was best to get Erythema, this weevil, back into Hackia stands after fires, because it would take longer for them to find their own way back. But in fact, if you've got Hackia close by, they actually recolonize pretty quickly. But it is one of those agents that can be collected pretty easily. It's a nasty plant to work on, but they can be collected and redistributed, particularly, again, as I say, after fires. Any questions regarding Erythema?
You just basically shake.
Yeah, I'll show you. I will go into that later. Okay, the next agent, again released in 1970, is this seed moth. It is incredibly cryptic. You can see it there on the left-hand side in that yellow circle. Very rarely do you see adult moths in the field. So it's certainly not something I would actually go out and try and collect loads and loads of adult moths. They lay their eggs right on the join. So the Hackia fruit essentially consists of two sides, and within each side there's a seed. So you've got two seeds inside a Hackia fruit, and they lay their eggs right on the join of mature fruits. And those little larvae burrow through that mature fruit into the middle where the seed is, and they basically eat out the content of that seed. And then, so they take 45 days to hatch, and then the larvae tunnel into the seed. The mature larvae then leave those woody fruits by a tiny, tiny little hole. And they drop to the soil where they pupate. And research has shown larvae destroy up to 62% of the seeds of mature fruits. The way that you would look for this agent is quite labor intensive, because it really is a tiny little pinprick of a hole that you see in the side of the seed. But we've had teams of guys, when we used to work closely with the Working for Water teams, they actually used to go out and they would collect loads and loads of Hackia fruits in the field. They would firstly just check. You firstly go and do a check. Are you seeing any of those tiny little holes on the fruit? And if you've got a team of 6 people that go out into the veld and maybe spend 15, you should find something within 15 minutes. If not, it's probably not there. And then if they found signs of it, they were collecting lots and lots of fruit. And then the fruit, when they left dry, actually split open. So then they were taking the half with the egg on it and going into the field, and I don't know if any of you have been involved in this over the years, but that fruit was then stuck onto a mature fruit in the field, and the egg would hatch and it would go into the mature fruit. So it's, as I say, it is incredibly labor intensive. They do disperse, and to be honest, I think probably it's best to leave them to disperse on their own, unless you've got a lot of time and a lot of labor to do the other process. But it it is possible to collect them and redistribute them. So as I say, the eggs can be collected on mature fruit, glued onto other fruit in the field for establishment elsewhere. But it's not an easy process.
And can you have both of them working together? So the first, I mean, do they?
When you say both agents?
Both agents, I mean.
Absolutely. I mean, and there are some areas where there might be all 4 agents. I think the bottom line is that, you know, the one isn't, so you've got one going for the immature seeds. There's always mature seeds that have hung around. So yeah, yeah. We then had in 2001 a stem boring insect that was released against Hackia, and everyone was very excited about this because, you know, this is something that can essentially kill the plant. It's unfortunately, I didn't bring any specimens with me, but the insect is probably quite hard to get an idea of how big the insect is. It's probably about 2 to 3 centimeters long. It's a nice longhorn beetle. And the adults lay their eggs right at the base of the Hackia tree. And those larvae then emerge and they tunnel into the stem, and they sort of tunnel up and down the sort of this part of the stem, and then they go into the root crown. And those larvae take 2 years to mature inside that root crown. So collecting the agent, and I did do a Hackia collecting trip for Aphanasium to Australia, and it is incredibly labor intensive. You firstly got to go and identify where the insect is. So you can see, often you see little bits of sawdust coming out of the base of the stem. You then cut the tree down, about so high from the ground, and then you've got to dig out, and you dig out very, very carefully the root. And what you have with Hackia is you've got a single taproot that goes down, and then you've got a number of these lateral roots. And you've got to be really, really careful when you're digging out, because often those larvae sit in the lateral roots, and it's very easy to break them. So the idea was then we would bring back sort of cut stumps or cut roots back to South Africa, and those would be hopefully with mature larvae. They're already in their 2nd year of development. And then the adults would then emerge in quarantine. We were able to do host spec testing with it, and we release adults into the field. I wouldn't recommend this as an insect for implementation, purely because it is really hard work. And unfortunately, whilst it can be extremely damaging, it can kill the plants. I think because of their long life cycle, they are very susceptible to fire. So a lot of the areas where we first did releases of this insect, the insects actually sort of fizzled out now because, you know, they just take too long to develop, too long to disperse. It is still in South Africa. It's still, there are some areas, Tatanes chlorophyno, there's quite a bit of it. So yes, if you're desperate for it, it is possible to be shown how to go and collect it. But as I say, it is incredibly labor intensive.
Sorry. So with the fact that it basically kills the plant, doesn't that actually add to the fuel loading indicators, because it's already intensive just to get it out again? So with the tree actually, with the plant actually dying, it contributes to the fuel load. That's why it comes to fire.
Well, yeah, I mean, you could be right. I think this, are you saying because then you've got dead Hackia rather than live Hackia? To be honest, I don't think there's enough dead Hackia around, thanks to this. And I don't work on this project, but I don't want to sound scathing about the insect, but I just don't think there is enough. It'd be wonderful actually if there was more dead Hackia, but I think the problem with Hackia, I mean, it doesn't, you know, you've probably seen in the mountainside, it doesn't all have to be dry to burn. Once the fires get going, I mean, they burn, and they'll burn through green live Hackia. And obviously the Hackia itself is, the seeds are serotinous. So those old seeds will hang on a plant until a fire comes through. And then they crack open, shed the seeds, they don't really have a seed bank, which is one good thing. All the seeds held on a canopy, but they're adapted for fire. They love fire, and you don't have to be dead to burn. So I don't think it's really contributing to the fuel rate. It's an interesting question, but yeah, I don't think so. Okay. Next up, a little flower feeding weevil, which was released in 2006, Dicomata rufa. And the adults of these weevils that I talk about, again, it's difficult to try and understand how big they are. They're pretty small. They're probably about 3-4 millimeters in size. Most of them are pretty tiny. And they lay their eggs up on the sort of new young growth, but also particularly on flowers. And the whole idea of this weevil was actually to try and stop the fruiting or limit the fruiting. And again, the larvae develop within the flower bud, mature pupae drop to the soil, and then they emerge as adults. This agent, although we had high hopes for it, hasn't really established very widely. It's established at a few sites up in the Southern Cape, but for some reason we've had real issues with establishing it down in this part of the Western Cape. It is particularly slow to disperse as well, and as I said, difficult. We've had difficulties with establishment, and its impacts are probably fairly minimal. Whether that's because it's just been localized or is localized or not, I don't know. So again, it is available if you go to the right sites to collect it, but I wouldn't recommend it necessarily for here because we as researchers haven't had success establishing it, and we can't work out why that is. Okay, the Hackia fungus. Now this is quite an interesting one because this is a fungus that actually wasn't brought in from Australia. This is a fungus which is native to South Africa. So Hackia is in the group of, it's proteoid. And the fungus was actually found on proteoid species. I'm not too sure exactly what species it was. And it was, it was obviously found close by on Hackia. It was looked into, discovered it was actually something that was native, but that could be developed as a fungicide. And essentially what it does is where the plant is wounded, these little fungal spores get in there, and then you get this sort of real gummosis, all this sort of gummy, sticky stuff coming out of the stems. And usually from above that point of attack is where you will get dieback. And yeah, you certainly can get death of trees from it, particularly if the fungal attack is lower down. But it certainly does stress the plants quite severely. It is also one of the agents that, whilst it can spread by itself, it's actually better if it gets implemented. So I don't know if any of you have done any work on Hackia, but you can obtain the fungal spores from ARC, PHP in Stellenbosch. Alan Wood actually does, and his group there, they grow it up, so to speak, and you can get little sachets of it. Alan will speak a little bit later, I think, about this particular species because it's something that in the past they always provided, and they provided it free of charge because we have been very well funded. Obviously, it takes time for him to isolate this fungus, to grow it up, to get it into the form where he can actually hand you little sachets. And I'll go into a bit more detail about it later, but that sachet you then mix with water, you wound the plant and you spray these fungal spores onto the plant. It's very easy to implement, but obviously the growth and development of that agent is quite expensive. So Alan will chat about it a little later.
Just to add to that, think about, before Alan talks to us, is he's basically trying to work out how you can charge and what is a reasonable way to recover some of these costs. And he's getting feedback from us because you guys have never really had to do this.
Well, this is what I'm saying. I mean, our time in the past has always been funded. Our, DFFE paid, it was part of the project. So biocontrol has always, in this country anyway, it has always been a free resource. And I think this is the thing, a lot of people have grown to expect that, well, they get something for free. And in a way, like anything you get for free, you don't always appreciate it. And, I know when I've done training in the past with groups and we've gone out into the field and we've actually spent the day collecting, people say, sure, but this is really hard work. And I said, well, yeah. And I think when you actually have to do it yourself, when you have to go out and beat insects down from a tree or dig insects out under bark, it is hard work. And, so it's very easy to sit back in an office and say, oh, please send me, I need insects for 5 release sites. I need 10,000 weevils. Well, it's taking somebody or lots of people a lot of time and a lot of effort, a lot of traveling. There's a lot of knowledge that has to go into it in terms of how you're keeping those insects while you're collecting. And so, yeah, I mean, obviously, as I say, it was funded in the past, it's not anymore. And this is why at the moment, I'm not involved at all with, really helping collect and release, because there are just no funds for that, which is it's a critical thing. Okay, so that's all with Hakia. I don't know, does everyone
I have a question about Hakia? It's just there's a species that's popping up in the Mossel Bay area and up into Rijkenbos. It's the Silicifolia. Are there any biocontrols for that, or?
I stand under correction. I don't think so. I can have a look in the back of this book.
It's starting to spread more in that area than it had been in the past. It was sort of a couple of trees here and there, but now we're starting to see it pop up in areas where it hadn't been before.
Okay.
I'm kind of finding it a lot in Ghana by where I live, which historically it was never.
I want to say that seed fit, the Erythena goes on to Silicifolia, but I'm not.
How does it look? I don't know the difference.
Silicifolia has got these
it's got
long flat leaves. It doesn't look like a Hakia really at all.
I've got photos on my phone.
So talking about weeds and identification of weeds. So this book, Leslie Henderson produced a book, Invasive Alien Plants in South Africa. So it's a really informative book. There are a few copies at the back of an older version of this. This is the latest book, which I think you can still get. I'm not sure who's selling it. I know they were also handed out free at one point, but that was in the days we had funding. But there are a few copies at the back of her older version of this book. So for weed identification, they are actually very, very nice. I'll check on Silicifolia later. I think that Erythena might go onto it, but I'm not entirely sure.
And the fungus only goes onto one of those, right? The fungus doesn't go onto.
Funny enough, I think there's another. No, I'm sorry, I'm talking about Aphanasium. So Erythena goes onto both species. Okay. There was another weevil which I didn't even go into in this discussion, which again is a seed feeder, goes onto both, but it's you very rarely see it in the field. Aphanasium, the stem borer, it could be that there are 2 different biotypes, but I know in Australia before we've collected off Hakia gibbosa, but there the Aphanasium tends to be a little bit higher up in the plant rather than in the roots. But yeah, they're going for both species.
And the
fungus doesn't, sorry,
yeah.
I think the fungus is just for gibbosa, but Alan will be able to confirm.
Yeah, I know it's only one of them, I just couldn't remember which one.
Yeah.
Yes.
We use the fungus on the silky monkey.
Is that did it work?
Yeah.
I thought it was only one of them, but I don't know which one it was. Okay.
So, well, as far as I know, it's for gibbosa, but maybe it does go. I'm not on that Hakia project, and I know very little about pathogens, so I'm just trying to explain to you what I do know, but it's very limited. But yeah, it could be that it is on both. I actually thought it was just on Gibosa. I mean, Sericea. But it's if you've used it, then yeah. Okay, so I'm going to move on to Australian acacias, if everyone feels happy about what they've heard in terms of Hakia. This has also been another one of South Africa's long-standing biocontrol programs, initiated in the 1970s. And it was one where we met with very serious conflicts of interest back in the 1970s, where when we were looking at potential agents, particularly in the line of seed feeders, we were actually taken to court by the wattle industry because they were not happy about what we were doing at all. And they were very, very concerned that if we introduced anything against any Australian acacia, not just black wattle, anything, that it would actually move onto black wattle and it would destroy the wattle industry. And back then, the wattle industry had a huge amount of clout. It was a very massive commercial industry. They were producing tannin for the leather industry from the bark. They were getting a lot of very high-grade pulp out of the wood, and that was being exported to Japan for top-quality paper. And over the years, things have changed quite a bit in terms of our relationship with the wattle industry. One of which actually is that we've worked, instead of working against them, we've tried to work with them, and we've been very open about what we're doing. We communicate openly with them, and we went through a stage where we were meeting with them annually just to discuss what we did and why we did it and how we did it. But essentially that conflict of interest that we met, and it was particularly with the wattle industry. Obviously there have been issues with people that grow oikons for firewood and Acacia saligna to feed their goats and such like, but those are smaller conflicts with not such big commercial industries behind them. But that has really led our choice in biocontrol agents. So, you know, very often, if you have an invasive plant that has no use whatsoever in South Africa or wherever it might be, you can really go guns and all. You can go and find something that is going to be particularly damaging. I mean, stem borers generally tend to be quite damaging, but you can find those agents. So we didn't really have that freedom. People often ask, well, why didn't you find something more damaging than a seed feeder? Well, the reason was that we actually had this gentleman's agreement, really, with the wattle industry, that we wouldn't release anything that would target anything that wasn't a flower or a seed pod. And that has continued to influence our choice in biocontrol agents to this day. And because of that, seed-reducing agents were really recommended. So to date, we have 10 of the most invasive Australian acacias in South Africa that have biocontrol that have been implemented against them. Not on this list are some acacias where there has been what I'm going to call spillover of our biocontrol agents onto Australian acacias. So, for example, Acacia elata, which is pepper tree wattle. You don't see here, but I know, and we knew in our testing procedure, that the agents would probably utilize them. Acacia stricta is not up there, but our agents use them. But these were the projects that we actually officially organized against those 10. And then down at the bottom, I've included Paracerianthes lefantha, or stink bean. It is a very close relative. If you look at the plant phylogeny of Acacia, and I'm not so South Africa has Vachellias and Senegalias. We don't have acacia in our native plants. They used to be called acacias. Australia had the battle, they won the name, even though this type specimen was a thorny tree. They won it purely because they've got so many of them. So we now have Vachellias and Senegalias. They are distantly related, not closely related. So in the past, people always said, oh, but what about our acacias? These things are going to jump onto our acacias. And actually, if you look at the phylogeny of the plants, they are very distantly related. But something that is a close relative is Paracerianthes, which falls into a family that is closely related to acacia. And we have biocontrol against that, which is why I'm going to cover it under the acacia umbrella. Okay, so here we have a number of, and I'm going to go through first insect gall formers for the acacia group. The first is a bud galling wasp, Trichologaster acacia longifolia, which was released in 1982. And this was also whilst we were trying to sort of iron out some of the issues that we were having with the wattle industry. But they did finally allow some agents to be released on things that weren't black wattle. So we managed to get permission for release of this little wasp. The females normally emerge, and males, but interestingly, they don't need to mate. They don't need a male. They can lay fertile eggs without any mating. And the females lay their eggs into very, very immature buds on the plant. And at the time they lay their eggs, it's impossible to tell whether it's going to become a reproductive bud or a vegetative bud. So you get galling on both, which is quite a benefit because it can actually stop growth from that point, or it can either stop vegetative growth or reproductive growth. And the gall is essentially a reaction to this insect feeding that develops. So it's this plant abnormality that grows as a result of the insect feeding. And the insects actually complete their development within that gall. I think probably all of you have seen, you probably all know longleaf wattle, you will have all seen the galls on the plants. If you cut one of those galls open, you can see there the insect larvae and a pupa. And when that development takes approximately a year within the gall. So they'll lay their eggs in November, December time, and throughout the whole year, that gall starts growing. And it's only then the following year, the insects, the wasps, adult wasps will emerge. This has been a very, very successful project, sort of average 95% pod reduction. Galls can easily be collected for redistribution during the late or early summer, during November time. But I think pretty much everywhere where this weed is, you're going to find the agent. It was very, very well redistributed in the 80s, early 80s. And I think that it disperses incredibly well. I don't know of anywhere. People used to use it as an identifying factor for longleaf wattle. They'd say, oh, well, it's got the galls on it. It must be longleaf wattle. So I think it's probably one where there is no need to do any implementation of it. It's sorting itself out. And yeah, as I say, this Acacia longifolia is not a weed that we get requests anymore for biocontrol. I think together with biocontrol and fires up in the mountains, it's pretty much got it.
Which areas does it occur?
Longleaf wattle.
Yeah.
It occurs right up into the Eastern Cape, so it's mostly coastal. But you do get it up into, well, I say coastal, up into the mountains.
Yeah.
But not sort of the really hot inland areas. So, and that'll be up Western Cape, Eastern.
You get roycrans as well.
Do they occur together? Yeah. Yes, they can. Roycrans is far more coastal.
Okay.
This is more sort of slightly more inland mountains where you'll find it. But you do get it right up into KZN. There are a few pockets of it. I know there are some small patches, Gauteng and Mpumalanga, but that gall wasp is pretty much everywhere.
And was it because of the good distribution that it spread so much, or was it because it is an agent that
So I think it's a bit of both.
Yeah.
I mean, funny enough, when you see these little wasps sort of hopping around, you think, gee, they're pathetic. I mean, they sort of buzz around on the table. They don't look as though they're going to fly particularly well, but they do disperse. So it was a bit of a combination. As I say, there was this big drive to get them. I mean, people would, back in the 80s, were collecting, taking bucky loads of boxes of galls and literally just chucking them as we were driving along. We'd chuck them out of the back of the bucky. So it was very widely, which I think helped a huge amount. Okay, staying with gall formers, and again, it's another Trichologaster species. It has exactly the same behavior as the Trichologaster on longleaf wattle, and this is on Acacia pycnantha, golden wattle. You guys at De Hoop will be very familiar with golden wattle. The distribution of golden wattle is fairly limited. There is some, probably its most easterly is up around Humansdorp. There's quite a lot there. There's some around Bot River. There used to be at Tulbach quite a lot. De Hoop still has a fair bit, but it's not as widespread as Acacia longifolia. This little wasp was first released in 1987, and it actually, the researchers at the time thought, well, it hasn't established. They had done some sleeve testing or sleeve releases where you actually release wasps in sleeves out in the field. They went back, and in fact, baboons had gone and ripped their sleeves apart. So when they went back, they struggled to find exactly where they put the sleeves, but they couldn't find any galls in the field. And they sort of felt, well, it actually hasn't established. So a second release was done in 1992, or second importation. And at that point, when the insects were taken out into the field, we realized, well, it's there already. So obviously it had established, but we just hadn't seen it back in the early 80s. But very, very similar mode of operation. The females again lay into the very young buds. Could be vegetative, could be reproductive, and it causes that gall to develop, and complete development takes place within that gall, from larva, pupae, and the adults. The adults actually chew a little hole through when they're ready to emerge, chew a hole out, and they fly off, and then they lay their eggs again. So the adults themselves don't feed. It's the larvae that are causing the damage by basically laying there, by using those buds. It stops the flowers from producing pods, or it stunts the growth in terms of affecting vegetative growth. Okay, this is the newest of our insect gall formers, and again, very, very similar to the two Trichologaster species that I've just spoken about. This is the one agent that took me a long time to get through the whole testing procedure. It was particularly for silver wattle, green wattle, and Bailey's wattle, which are mostly problematic upcountry, although there are some small pockets of silver wattle and Bailey's wattle around here. It's in a different genus, so instead of being Trichologaster, it's Perinampella, and it was released in 2023 in the Stellenbosch area, and it established, and we actually now have there's a nice little population of it. And so it was, I think I said that it was released at 9 sites and is established at 5. And one of those had established quite nicely. So it was enough to do a small collection for release, a new release last year. It's one of those that is desperately in need, or it will be desperately in need of implementation along the line. And there are a lot of people at the moment that are crying out for this insect, but I think we can't do any sort of large-scale implementation of it purely because it's only established at so few sites. So something I was saying to Aileen a while back, that obviously whilst we would really like people to get involved along the line, at this point it's still it's still very new, so we don't really have enough resource for 100 people to come and get bags and bucky loads of balls.
How long does it take? So how long do you have to wait? What stage do you feel comfortable with? No, so you can't diminish that.
Well, I think at this stage, what we're trying to do is set up more nursery, what we call nursery sites, because you can't mass rear this. It's really, you know, there is a process in biocontrol where you can mass rear insects. So in other words, under laboratory or shade house or whatever conditions, you can actually bulk up insect populations. It works particularly well for water weeds. So you can keep them in nice ponds, you can rear up insects. For when you're working on trees, it's just not really suitable to do it because you need to have massive trees. So for us, what we do is we have nursery sites, and we call them nursery. So it's field sites where we know that they're safe, where the insect has established, and we allow the population to build up. So, I mean, ideally, now in this year, depending what we see towards the end of the year, when the galls start becoming obvious on the trees, is if at those 5 sites where we had establishment, there's enough to do a collection to release elsewhere, I would like to get nursery sites, so like for you guys, to get a nursery site set up. But again, you know, one needs to have the trees. So we need to have either Acacia dealbata or Acacia decurrens or Acacia baeliana.
What's the common names of those?
So it's silver wattle,
Yeah.
is Acacia dealbata. Decurrens is green wattle.
Current
And
greenwood.
baeliana isn't really, they call it baby's wattle. In America, Australia, they call it Cutamandra wattle, but it's, yeah, so, yeah.
There's green, and it's quite difficult to identify the green one. I struggle.
But you have got green here.
We have got it? We think so.
Yeah. And look, I mean, obviously for setting up the nursery sites, and I do go into it, I'll go into it a little bit later, it's really important to have something that is safe, but also something that's relatively accessible. You don't want to be going on a 10-hour hike to have to go and collect your agents, because often you have to carry stuff with you. So, you know, you want something reasonable. You don't want it so accessible that everybody can see it and go and steal your resources before they really bulk up. But yeah, so it's, accessibility is important. And the trees just need to be growing nice and lush. You don't want old dying trees. So you want those sort of weeds that you don't really want to see. You want that type of stand for your insect nursery site. Okay, so that is the last of the wasp gall formers. Anyone got any questions on what I've gone through so far?
Anyone need a break?
I was going to say, do you want to have a break?
I just have one question. We've seen, I don't know, it's not those ones, but we've seen in field the Terminator bugs. And they're just targeting one species, which is the bulb pussy, which is indigenous. So I have a picture of how they look, but
I don't even know what a Terminator bug is.
I have a picture of it. Yeah, the assassin bugs.
Okay.
So I just wanted to find out whether are they also a biocontrol or are they
That'll
just,
be something native, so it'll be probably.
because
And look, I mean, we've also got introduced insects that we have got nothing to do with. It's not been our part of our, but it happens. I mean, I know the gum trees, for example, and this polyphagous shot-hole borer that's running riot in South Africa. So insects do come in. I mean, just like plants come in by mistake. Insects, pathogens, they all come in, you know. The thing is, actually, if you look at our border controls in terms of people coming in with things, they're not as stringent. If you go into Australia, New Zealand, those countries they've got incredibly strict regulations at airports, at ports, you know, so. You know, if you go in with a pair of hiking boots into Australia, they will take those hiking boots and they're going to take them off and sanitize them and bring them back. So they're obviously trying to avoid, not to say that it stops everything, but it just helps. So yeah, I mean, I suspect, unfortunately, wasn't introduced as a biocontrol agent. And some things also have these outbreaks. So you'll have outbreak years, because the climate will be right or whatever, and then they just go bananas on a particular plant. But yeah,
Really no bananas.
yes, but not a biocontrol agent. So I don't know if anyone wants to stretch their legs now.
Stretch legs? Stretch
Yeah.
legs, yeah.
Yeah.
How do you think?
I can do it.
Yeah.
Oh, there. Maybe to the side. It's better because it's a bit lower. What is that?
Kan ek net vra as julle plaas met julle koffiekoppies, moet net so op die tafel te sit. Nie buite nie, asseblief. Ons het Bobbiane.
Dis reg aan die hoeke.
Reg. So as jy net plaas jou koffie en jy laat, sal jy net buite
Fascinating, really great. Good.
I am intrigued.
Because they just uncovered and they eat the whole thing and it's nothing left. And it's just because I think it has like a multi, if you break the plant, there's like a multi substance. Okay.
Yeah, so it's poisonous plant.
Yes,
Yeah,
okay.
so
And it has that latex,
Yeah,
Yes,
yeah,
yes, yes,
yeah.
Yes, okay.
like I just told them we have our own insect
You
eating
enjoy
everything
it,
out.
What is that?
I don't know why, I can't tell you what I did, but that made a difference.
I think who else we. Did you find some break on something?
I didn't really look.
Oh, no.
No, we just walked up the bank here. Okay, so there's now another group of ball formers, but these are all little midges, and they are really tiny, sort of thing that you just swat out of the way, they're so small. So the first one is this Dacinura dealsy, was released in 2001 against Acacia cyclops or roicrons, commonly known. And with these things, the females, instead of using the little growth buds, actually lay their eggs into an open flower head. So I know initially with this, we released the insect, it took a little while to go, but people said, oh, your agent's not working, my trees are still flowering. And we'd say, well, that's the point. Trees need to flower, plants need to flower, because the insects will go and they lay their eggs into that open. So a flower, an acacia flower, is actually a flower head made up of 30 or 40 individual little flowers. And the females lay their eggs into those individual flowers within the flower head. And you get what you see up there on the left-hand side or down there. you get what's called a gall cluster. So much as people refer to that as a gall, it is actually a gall, it's a cluster of individual galls. And if you want to get really technical about it, each of those individual galls can have up to 16 chambers with little insects inside them. So you can get quite a lot of insects within a single gall. So this agent, very soon after release, started spreading very, very quickly. I mean, it was in the Stellenbosch area. Within a year, it had gone right up to Bonaclip Bay up on the west coast. It spread, so it spread very, very fast. And one of the, well, there's several reasons. A, it's very light, so it gets carried up on the wind. But secondly, and perhaps more importantly, it's multi-volatile. So it has several generations per year. And it's allowed to do this because the flowering of Roykrans can actually be quite extended. And you will get these sort of galls forming slightly different times of the year, allows it to go through several generations a year. So populations can build up very quickly, and as a result, spread very quickly. So what you would look out for if you're looking to see whether you've got this or not, don't look for the midge in the really battle, look for the galls. And the galls are very distinctive. You've probably all seen them. And you can pretty much see them on the plant at any time of year. It's normally sort of towards the end of the year, early part of the year, that you'll see green galls. But otherwise, you'll see the old brown galls. And the whole development takes place within the gall. Again, so eggs, larvae, pupae, adults emerge from the gall. Whoops, went the wrong way, sorry. Okay, then this next flower galling midge was released. Well, I say released. It was a little bit of a sort of, perhaps I should say, an accidental release. But fortunately, all the host spec testing had been done, but the horse bolted before we got final permission. But it has been an extremely successful midge, thankfully, and highly host specific to black wattle, and funny enough, it will go on hybrids of black wattle and green wattle. So quite a lot of the wattles, like black, silver, and green, the ones that look very similar, black wattle, black, silver, green, and Bailey's wattle, they all have these bipinnate leaves, and they all hybridize. So quite often what we have in the field is it's a bit of a mishmash, and this insect will use a hybrid of green and black. It won't go on to green wattle, but it'll use a hybrid, which is quite, and as will the previous one on the one on Roykrans will, a little bit more sort of generalist amongst the acacia group, but it will actually use a hybrid of black and green wattle as well, which is very interesting. And again, these females lay their eggs into the open flowers, not into the buds. And the flowers, instead of producing hundreds of pods like you see up there, produce these clusters of galls. And I've done quite a lot of work over the years looking at how seed rain has been impacted on black wattle. I was chatting this morning and I was saying, you know, it's all very well knowing every year, you know, so much percentage of the seeds taken out, but actually for you guys, for guys that are managing weeds, what is really important is how much seed is being produced each year, how much seed is in the ground. And I'm not a botanist, but I've kind of become a botanist over the years because I just feel it's really important to know that aspect so that you can then say, if you know how much seed was coming down before you introduce your biocontrol agent, and you know, some years we know it fluctuates over the years. Some years are particularly good rains, other years might be drought, not such a good year. So you do get these fluctuations, but they're generally fluctuations like I showed in that very first graph. It's at a high level. So seed of black wattle will probably sit anywhere between 5,000 and 10,000 seeds falling per square meter over the years. And we looked at that for about 9 years before before releasing this biocontrol agent, or before it's really started taking off. And now we're finding seed numbers are down to between 3 and 20 seeds a square meter. And that has been consistent since having this biocontrol agent in place. So from that, we can actually deduce, okay, this agent is doing a great job. Always the Achilles heel with so many of particularly the Australian acacias is the seed bank. So, you know, much as you're doing this wonderful job above ground, what's down below ground is there for pretty, not eternity, but it's there for a good 50 to 100 years. They have very, very long-lived seed banks. Those seeds can lie dormant. They don't germinate spontaneously, so they can just lie there until the right conditions suddenly come about, and then you know, you can have a fire, they won't all germinate at once. Some will, some won't. And they've got this sort of inbuilt dormancy that is quite hard to break. But again, I think it's the whole thing of what can a seed feeder achieve? So, I mean, I would say, you know, if you're curtailing seeds, so if you've got an isolated tree, for example, that's producing thousands of seeds, those seeds are spreading away from that isolated tree. Some are accumulating in the seed bank and some are popping up as new plants. Those plants end up growing up, they produce their own seeds, and that's generally how invasions start moving. But if you can reduce that amount of seed that is being produced by that isolated tree, you're obviously reducing the rate of spread, you're reducing what's going into the soil for accumulation. It's got to. It's one of those really difficult things to try and prove scientifically. You know, a lot of people say, well, you need to prove it. I don't have the wherewithal to really devise those sort of experiments to prove that. But, you know, theoretically speaking and logically, it's got to have some impact. So, you know, when you're sort of stopping seed rain to the point where you've almost sterilized a tree, you know, when you're getting 3 seeds as opposed to 10,000 seeds every year, that in the long term, maybe not my lifetime or any of your lifetimes even, but, you know, along the line, and I think this is often where we've got to say, well, it's not just for the here and now. These are for future generations that we're doing this for, and then I think then we can say we've achieved something.
At which stage should one collect the galls?
Okay, so these galls generally need to be collected in winter. That's when, so unlike the previous midge, this midge actually doesn't complete its whole development inside the gall. So the larvae develop inside the gall, and just before they're ready to pupate, so you can see in that middle picture down at the bottom there, you see this orange, sort of bright orange, fat little larva. They actually leave the gall and they drop to the ground, and they make a little cocoon out of silk and they pupate in the ground. So what you want to get is you want to collect those galls just before the larvae leave them. So a lot of people I've spoken to many people, oh, they saw galls, they collected them, they took them to their farm and they didn't get establishment. So when did you collect them? Oh, well, we collected them in February or March. I said, well, that's why. All those larvae in that gall would have died because they would have been too immature. The gall then ends up drying out. So the timing for collection of this is really critical. And as I say, normally and it depends obviously slightly where you are in the country, but we generally work on sort of mid-June through to mid-July. What you can do as a very sort of quick check, you can get a subsample of galls and you can put them in an ice cream tub. Just spray them with a little bit of water, and what you should see, if the insects are ready for emerging, you'll see little orange larvae in the bottom of that ice cream tub. Likewise, you can get a plastic bag, although plastic bags tend to encourage emergence. But if you see larvae coming out and you see them in the bottom of the bag during the course of a day or two, the galls are probably likely to be ready for collection and redistribution. What we initially were doing with this agent was checking under a microscope. but obviously we don't all have that luxury. You can normally see them, the larvae, you can tell when the larvae are ready. So I know the implementation teams were working very closely with us as researchers. So we would say the timing is right. One of the things we tried doing for getting the agent spread countrywide and relatively easy was that we put the galls onto squares of old carpet. We used to call them magic carpet squares. So the galls would be put onto these squares of carpet. We used to just give them a little spraying of water every now and again. You could refresh the galls even. So what the larvae were doing is they're coming out of the gall and they were going into the carpet and they were pupating in the hair of the carpet. And then you could then pack up those carpet squares into nice boxes, let them dry out if they were wet, pack them up, and you shipped off a box of magic carpet squares. And then people would go and pin them onto their tree trunks. And the adults would emerge out of their little pupal cocoons. So it was a really neat way of redistributing this agent. The only problem we did find was quite a lot of carpet is treated nowadays for insects and things. So, you know, you got to be, it wasn't kind of like a one size fits all type thing. People tried doing it and it ended up being, you know, a lot of the larvae were dying in the carpets. So in the end, we just said, okay, look, rather let's send you a box of galls. It's obviously more expensive shipping that way, but at least you're guaranteed of getting nice fresh galls that you have to, but the timing then, you don't have a lot of time between collecting your galls and releasing your galls. And that's what we always, so you can keep them cool, probably 3 or 4 days. So, you know, the one thing, courier services aren't always that reliable. So you send galls to outlying areas of the country, it can take 4 or 5 days. So we've always said, you know, if you receive those galls, try and release them the day you receive them. If you can't, keep them refrigerated, keep things cool. And I will go into this a little bit later. But yeah, so does that answers?
Sorry, this is probably a very stupid question, but you guys haven't found anything that would feed on fallen seeds like that would listen your seed bank? No,
somebody asked the question earlier. There, I can't remember. Oh, it was you asked me this morning. Sorry, whilst we were having breakfast. Unfortunately, not. Not for acacia. There are for some of the agents that we have on Prosopis, which grow up in the Northern Cape. They'll even find seed in sheep dung and they use it. But acacias, no. The reason being, and I will get onto it now, actually, is that, this is not agent, but the seed feeders generally will go for green seeds. So they need that green, they need the green pod so that the larva can penetrate into the pod. The seed needs to be nice and green and fresh and new for them. Once the seed hardens, it becomes difficult then for the and I think that's, you know, that's obviously what you're dealing with. So besides then looking at pathogens potentially for on the ground, there's nothing that I'm aware of. And even, I mean, in Australia, obviously there's not really that amount of seed that drops, but rodents eat it. And rodents do quite a lot of damage, particularly to, say, Roikron seeds. But they also do put them in caches. So, I mean, it's yeah, but unfortunately nothing available for seed that's just on the ground or seed that's in the soil. There, you've really got to depend on natural rotting processes, which they certainly do take place, but it's just slow. It's much slower, unfortunately. Okay, so the last of the flower galling midges, and this is the one that we really struggled with getting this thing established. We first got permission to release it in, I think it was 2000, it was either 2016 or 2018, I can't quite remember. But this is the one that we got established, and then our field nursery site burned down. And then despite 2 or 3 importations of this midge after that, we just could not get it established. It was very difficult. And I think part of the problem is that many of the acacia flowers are very susceptible to high winds. Well, firstly, the plants themselves. If it's a dry year, particularly dry year, they just don't really produce many flowers. If it's a good, nice wet year, well, they flower profusely. And then if it's very windy or if there's hail, the flowers all blow off and they drop off. And we just, yeah, it could have been that where we had real problems, but anyway, we finally now got it established. And funny enough, because it has the same host plants as the little gall wasp that I showed you in this last picture. Oops. That. Exactly the same host plants. And we released them into the same spot. And last year, I was really excited because I saw, wow, there's all these galls of this new agent, but then we struggled to find the galls of this, because it's almost like the one that displaced the other. In Australia, you do see them on the same tree. You see the two together. And I think here in time, and in fact I did on further searching, I have found this midge still in Stellenbosch. It's still there. It was just lower numbers. Again, so we're trying to establish nursery sites for this, but again, it's going to take a bit of time. But just like the other gall midges, they lay their eggs into the open flowers. The flowers don't produce seed pods. You get that gall-like formation, and the whole life cycle takes place. The whole life cycle takes place within the gall. And even when the gall dries out, the little insect cocoons are still inside there, and the whole gall drops to the ground. But they actually stay in that. They don't leave the gall and pupate elsewhere. They stay within the gall. What else did I want to say about this? So here again, we would generally do the collection of this midge probably when the galls are still hanging on the trees. So it's a little bit more difficult, but it's better to collect this, say, in November, December time. So the galls are mature, they're on the trees, they're quite easy to mature, but they're just starting to turn brown. And then those galls would be collected, and you can maintain them in little gauze bags outside, or you can go and release them immediately. But the females will not emerge from the galls until about May, June time, which coincides generally with the flowering of the host plant. So you can look at it either way, but it does make it quite a nice agent for redistribution, in that you can wait until the galls are nicely dried out, leave them outside, and then just send a little box of galls. You've got more time to play with once you've got the galls. But as I say, it's a very new agent and not quite ready for large-scale implementation. We're looking more at setting up nursery sites here for this one. Okay, staying with gall formers, but moving on to pathogens, which, as I say, I really know very little about. And I think all of you here are very familiar with this rust fungus on Port Jackson. It's been, again, a very long-standing project. The rust was first released in 1987. So Alan Wood is now working on it. Back in the day, it was Mike Morris, and in fact, it was named after him. Ultimately, it was initially given another name. It used to be called Eurymycladium tipperianum, and then more recently it was renamed. And this rust actually specifically goes for, they like new growth, new fresh growth. So they will actually affect both reproductive and vegetative growth. And unlike the insect gall formers, this gall former can actually kill trees. But it does generally take quite a bit of time. It's not going to arrive on the trees and kill them immediately. So you can look in 5 to 10 years for a plant that's heavily galled to eventually succumb to the effects of the pathogen. But what has happened with this is that in many of the areas where it's been particularly abundant and effective, we've had decreases in populations of the weed, which is obviously an ideal scenario. Because it is so widespread, the spores get easily carried on the wind. There is no need any longer for redistribution of the pathogen. Okay, and then this is a more recent pathogen. Some of you may have seen it, others maybe not. It's named after Alan Wood, Myromycladium woodii, and it is the rust fungus on Paraserianthus lephantha, was released in 2016. And yeah, it has been incredibly successful. It does kill trees, and as Alan told me on Friday, even isolated trees. So they don't have to be, I mean, you can see there, you know, the sort of before and after type pictures. So it does, it targets trees not only in dense populations, but very isolated ones, and it's dispersing naturally, and it's doing very well on its own. Because I said, well, do we need to get hold of spores from you? And he said, no, it's doing its own thing, which is great. Yeah. And then just quickly, because these aren't gall formers, but just to say that there is also another fungus which is available from ARC, from Alan, called stump out. Some of you may have utilized it. It is very effective on those trees that don't sucker. So you can use it very effectively on black wattle or golden wattle, but it doesn't work well on Acacia melanoxylon, blackwood. It doesn't work well on silver wattles. So any of those wattles that send out the rhizomes and shoots, not rhizomes, but the shoots, it just is not very effective. But for black wattle and golden wattle, cut the stump and you apply it to the cut stump. And
it's like an alternative to herbicide.
It's an alternative to herbicide. So for a lot of people, you know, that are wanting to be completely green, they don't want to use herbicides. It is available, they can make it, and it's I think one of the problems when we were working for water and spray teams, one of the things that came up was people don't really want to be carrying both. So if a spray team's going through, if you've got a clearing team that's going through an area and they're treating cut stumps with whatever gets used poison-wise, then they've got to carry something, you know, so they've got this weight of something else. So this was one of the things. So unless you're going through a monoculture of black wattle, then they felt, well, it's just something extra to carry. But sure, it's cheaper than herbicide, and it's kinder of the environment.
So, do you can say the time frame of the before and after?
All I know is I think this would have been released, so we'll have to ask Alan this afternoon. I cannot be sure, to be honest, but it's, I mean, it wouldn't be months. You say it would be months.
Yeah.
So you've had the experience of when he told you it's gonna kill plants, and you're a bit sort of apprehensive.
Yeah.
And obviously a few months later the tree was dead, or so I think it depends on the size of the tree as well. So if you're dealing with a sapling, you know, as opposed to a massive big tree, that might determine how long. But I mean, we can ask Alan. I'm not too sure what the time frame would be.
Does climate have a big impact on the effectiveness? Again, it's not an Alan question in terms of the.
As I say, what little I do know of pathogens, I would say yes, because I think they often need. So they often say if you're going to apply, I know hack attack, for example, they say if you're going to apply those fungi, make sure that it's quite moist, because I think fungi, and your pathologists, maybe they do better under moist conditions. I stand under correction. I'm not too sure. Okay. Right, now onto the seed feeding insects on acacia species. So when I first started working back in the 80s, this was my project. And we now to date have 5 different acacia, 5 different, they're called Melanterius, the genus is Melanterius. So we have 5 different Melanterius species. that have been introduced against 10 acacias. So what happens, some of them, so the one up at the top is specifically against Acacia longifolia. There's one specifically on Acacia melanoxylon. There's one that's on Acacia cyclops that actually also interestingly uses Paracerianthes leuphanater. So it will use stink beans, it's exactly the same species. There is this one that's on Acacia saligna. And then there's the one at the end there, which actually is primary host is Acacia mearnsii. But, and I initially thought that it was maybe biotypes of the same species, but I've had genetic work and whatnot done. It's the same species that uses silver wattle, green wattle, golden wattle, and then another wattle, Acacia podularifolia. I don't know if you know it, it's got very grey, soft leaves, and it also uses Acacia baileyana. So it's got a suite of other hosts, but within the acacia, the Australian acacia group. So it's not when I say other hosts, it's not moving on to other hosts. So all of these weevils, again, they have a very similar behavior. And the adults feed, initially, they feed on the green seeds. And by feeding on those seeds, they're actually killing the seed. So the death of the seed comes in two ways. Firstly, by the adult feeding. And secondly, they then lay their eggs onto the seed. And those eggs then turn into larvae. The larvae tunnel into the seed. And the larvae eat out the whole of the seed content. Usually it's just one larva, one seed. But what can happen if the seeds are particularly small, they can move along the pod and they can eat several seeds within a seed pod. And when they're fully developed, they go through several different, what they call larval instars. When they're fully developed, they chew their way out of the seed pod and they drop to the soil and they pupate in the soil. They have only one generation per year, which ties in with the time that the seed pods are maturing, turning from green to brown. They disperse very slowly, only about 2 kilometers per year. And this is why they are actually particularly good for collection and redistribution. And I think it's quite relatively easy to do that. Damage levels vary. It's difficult to put a set number here because we're dealing with so many different acacias, and it varies depending on the pod production or the flower production, because it's quite erratic with acacias. So it's quite a difficult thing to really get a handle on. And the effects of the weevil can be very influenced by things like site disturbance, clearing, fires. And also, as I say, because of plant reproductive phenology, some years you get particularly good podding, loads and loads of pods, so populations can build up, and then the next year, very few pods. So suddenly you've got very few pods and a very big population, so your populations, so they tend to sort of, they go a little bit up and down in terms of their population. They are reasonably easy to collect. We call it shake sampling. I will show you a little bit later. And then if you're wanting to see whether you have these agents present or not, you can pretty much go at any time of year and find an old seed pod, whether it's on the tree, on the ground, but it's probably better to do it when those seed pods have just matured. So I would say December, January time, go and look, even when the seed pods are still hanging in the trees. But if you're desperate, you can look around on the ground and look for these. That's a very classic sign that you have the seed weevil present, is that little round hole. And whilst there are some other secondary, so there are moths and things that use these pods that have opened. The easy thing also is to open up, if the seeds are still inside the pod, you can actually open up the seeds and then you'll actually see, I don't know if I've got a photograph here, no, I haven't. But you can see the signs of damage to the seed. Either you might see the seed inside with the hole in it. That's very classic of this particular insect. You might even, if you collect pods in, say, November time, and you open them, you might even find the larvae, like you see there, still inside the seed. So But the classic is seeing those holes.
And it's only this that made those holes, no indigenous
No.
streets. Those holes is always going to be, it's not a, no.
Yeah, I mean, as I say, sometimes, and they're not big. Some people cut, and it is a very circular. It's a nice, neat little circle. It's not a messy. I mean, sometimes you might say a moth might have been in those pods. I mean, I know they're native moths that use them, and when they chew their way out, it's not a nice, neat hole, not like that. And they are small. I mean, we can have a look. Certainly, we can look around here. I've seen them on the blackwood trees here.
Yeah.
So this is quite interesting in that we initially thought that the adults can live for up to 2 years. But what happens is that when they're in the soil, so we always thought, well, they pupate in the soil and they emerge within a year. But actually what happens is that there's a portion of the population that doesn't emerge and they stay dormant in the soil, and they can stay dormant in the soil for up to 3 years as larvae. And it's actually quite a good sort of life history trait to be able to do that, because that way, if there's a fire or whatever, I mean, some years, for example, because of, say, for example, if you look at black wattle, we've got this midge that is doing extremely well. So some years there are very few pods. And we always think, well, it's going to be to the detriment of the weevil. The weevil populations are, and whilst they have been affected, because of this delayed emergence, I think they are able to emerge at any time, and there are always then pods that they can capitalize on at the right time of year. So it's quite an interesting life history trait. Okay, and I've said you can collect adults relatively easy by shaking the trees, and I'll go into collecting methods a little bit later.
So what about certain populations of trees developing resistance to some extent? Because what I see with Roy Crown, specifically with the weevil, is that on a single farm, very dense stands of similar age Roy Crowns, and one area quite prolific, and I mean they go in, the woodmakers have cleared the area, and there's like one little Roy Crowns every 3 meters that come up. There's just no seed, and you find all the seed balls. I counted actually 19 out of 20 seeds have been killed. But then 3 kilometers further on the same farm, there's virtually nothing. So that's not even 3 kilometers, maybe. So do you see resistance in the trees, or is it just a matter of...
Yeah, so that's an interesting question. And funny enough, I haven't noticed it with the seed weevil, but what I have noticed is that with that midge that's on Roy Crowns, you can sometimes get trees side by side. And one tree will be very heavily galled, and the tree next door, and I don't know what, and I don't know if it's resistance per se, because I don't know if you come back a year later whether that tree has galls or not. But something, it's a strange thing. I don't know why some trees are preferred and others.
Is there evidence in the literature of there being resistance and needing to get new? I mean, in general, is that something that happens, or?
I think it probably could happen, because I mean, obviously plants are always.
Yeah, so I mean, with other biocontrol agents in the forestry industry, it has happened that if you have a single introduction of a biocontrol agent into the country, but you've had several introductions of the host plant, then you have a very nice genetic spread of the host plant, which enables it to make it more resilient. And then if your source population for the biocontrol agent is genetically limited, it makes it less adaptable to the variety of genetic variety of the host. And I think this is, so with Relay now, what we've done is she would bring us weevils for Roy Crowns from Stanford, for example, and we would collect some here in Stillby that she would take back there. And we try and even in my area, the plan is now that, I mean, unfortunately, that window is so short, it's 3 weeks that you've got, to really spread around, so I'm planning my whole year around this now. Because to me, as an extension officer, I don't have an operational budget, but this gives me a tool to actually try and get land owner permission. So if I come and tell them we want to release the agent there, they're very happy.
Yes.
And even if you just do a single release there, they're still happy. They don't, anything you do is I mean, obviously we ask that where we release that revised recommendations is that they must sort of guarantee us that they're not going to cut down those trees or have a controlled fire there in the next 2 years, so that you at least have your population established. But there's no point in releasing stuff if it's going to be cut down.
Sure. I mean, it's interesting. I mean, they refer to them as genetic bottlenecks.
Yeah.
You know, and I haven't actually really been aware of it with any of the acacia insects. So it's quite interesting that you're talking about this. And in fact, if I look at the midge on black wattle, I mean, because a lot of people say, oh, it's bad, and you've got this. But in some cases, it can also be quite good, because you're sort of pruning out the weak sick layman lazies, and you've got this strong population. So certainly with the Dacenura midge on black wattle, that all of what we have today came from a very, very small founder population, because they were essentially escapees. So
well, they might have been strong.
Not my project, but
strong ones escaped.
Yeah, so for the right grounds, I don't have any evidence that it's really the case. I just work on the assumption that to me that is logically that's type of stuff.
I mean, it'd be interesting to see whether you have any success with that. I mean, it's a nice experiment to do, just to see.
To really
As I say,
something like that is very difficult.
I've seen it with the midge, but not with the weevil. So it's interesting. Okay. So that's now it in terms of what is available for Australian acacias. And so does everyone feels happy? Okay. So now moving on to Sesbania punicea. This is a project I worked on a little bit when I first started off as a technician at UCT. And this plant, unlike the previous two that we've spoken about, Hackia and acacia, this is a South American plant. It's not a plant you see abundantly anymore, and that is thanks to biocontrol. And people still complain about it, but they complain about trees that vary from sort of this height to this height. And I know when I started working back in the 80s, we used to walk into forests of Sesbania that were as tall as a ceiling up there in the sort of Cedarburg area. So, and big, really big, you would get big, healthy trees. So there are 3 biocontrol agents that have been released against Sesbania. The first is this little flower bud feeding weevil. It was released in 1981. And the adults feed on the leaves. So one of the ways that, and probably the easiest way to tell whether or not you've got this weevil, and sorry, it's a really bad photograph, but the photograph on the left shows what we call typically shot hole, that sort of shot hole effects on the leaves, and that's from the feeding of the adult weevils. You can, if you know what you're looking for, look in the flowers. So the times that they produce their flowers, you can look in the tiny buds, you can see, but it helps if you have a microscope, you can see the larvae. So they're mostly damaging, they damage the flowers and stop the flowers from producing their seeds. They complete the whole life cycle within the flower bud, and the life cycle is really rapid. I mean, I think it's probably less than a month, and it's literally as days. They can work through their life cycle, and so you can get multiple generations during the flowering period. They are very widespread. So Sesbania occurs in most of the, well, other than the dry provinces, but it's in most of the provinces, typically along riversides where it's sort of a bit moist and wet. That's what they like. But this weevil is pretty much everywhere. So I would say there is no need for collection and redistribution. You often get it. If you ask for other insects, this will be in there with collections of those, but chances are you have it already. Then the next weevil is a seed-feeding weevil. That previous little weevil is tiny. It's probably a couple of millimeters, very, very small. This is a much bigger weevil, probably between 0.5 centimeter to 1 centimeter. And like the acacia weevils, it feeds on the green seeds, but also lays its eggs on the green seeds. They develop within the seed, eating out the contents of the seed. The larvae then find their way out of the seed pod, they drop to the ground and they pupate in the soil. And a bit like the acacia seed weevils, they can be collected quite easily by shake sampling. And then we have this really pretty weevil, which is quite a large stem-boring weevil, released into South Africa in 1984. The weevils lay their eggs onto the plant stems, and then the larvae actually feed under the bark, and they will often ring bark firstly, and then they actually go down, and they go sort of up and down the lower part of the stem, causing that sort of damage that you see. And they will actually cause death of the plant. Yeah, so usually they will go for you can't put them on little spindly Sesbania plants, so they need to have reasonably, I would say probably something about 2-3 centimeters in diameter. Thank you. They would need, because they need something substantial to tunnel into it. And for collecting these, the easiest way actually is to hand pick them. So because they are real, you get what they call well, for us anyway, we call the clingers and the droppers. And there are some insects, when you bash a tree, they drop, and others cling. Well, this is a clinger, so you can't, if you try and shake them out of the trees, they hang on for dear life. And they're impossible. Even when you try and pick them, they tend to cling on to the...
Don't give it at first a light shake.
You can try, but it's amazing. It's almost like they feel you walking up to the tree. They pick up very, very, I mean, I've had it where even putting out a beating sheet, I haven't even touched the tree, some insects will fall. And others you can see straight off so it's, and that's why I say with these, you've really got to look for them. And look, they are pretty beautiful, and they're easy to see. So there you would just pick them and put them. We use little mesh bags, but you can use a pillowcase or something that you can keep them in there until you go to release them. And then most effective control of Sesbania, you need to have all three of these agents working together. There's been a lot of John Hoffman at GCT's written a lot of papers on Sesbania control, and certainly they found that best control, most effective control is all three of the agents together. Okay, now on to Leptospermum, which is a little bit of a problem plant, literally. For us, particularly as biocontrollers, because something that we found is that often the distribution of Leptospermums often overlaps Port Jackson. They often grow together, and where we found Port Jackson sort of thinning out because of the fungus, this thing is just taking over, and it is becoming very abundant, very prolific, and it just produces masses and masses of seeds. And unfortunately, from a biocontrol perspective, we have really had not a huge amount of success with biocontrol. Funny enough, the first, well, it wasn't actually the first, so this leaf mining moth released in 1996. So the larvae of the moth actually when I say mine, they kind of tunnel through the leaves, and I don't have a picture at all of what you would look for, but you often get either puffed up leaves, or you can actually see the little white mine or the tunnel. If you look at a green leaf, you can see, but the impacts of the moth have largely been negligible. The trees just seem to keep on going quite strongly, even with the damage that they inflict. The second agent that we introduced was a bud galling midge. So it looks very like it's in the same group as the midges on acacia, but obviously very specific to leptospermum, introduced in 1994. And initially, so this actually lays its eggs in the little growth buds, the reproductive buds, and it stops them from either growing or producing the little seed capsule. And initially it looked really promising. We were getting really high levels of damage. Those were the initial reports. And even though we know this midge suffers incredibly high mortality, there was still plenty of it around. They're still on the plant, very evident on the plant, but damage is highly variable, and it really sort of fluctuates around negligible to sort of reasonable. But where both the agents actually have their highest level of impact is on the small seedlings that come up. So there it can be quite damaging. And I think it's because obviously you get this little single shoot. So if it gets defoliated, or you've got these little galls on the growth buds, it's putting quite a lot of pressure. But for a big tree, really they're not having any impact. And there was a lady who was working on this project who's left South Africa, now living in Australia, and she had a student, a PhD student, who was looking at potential new agents for leptospermum. Unfortunately, the PhD went a bit pear-shaped, and the student pulled out of it, and so there's actually no one working any further on it. And yeah, we've really struggled with it. There have been a number of agents that we've looked at. We've tried collecting the scale insects and things like that. We've looked at. It's been a difficult target plant, which is unfortunate, because I think it's a plant that
it's terrible.
People are, yeah, and it's just getting worse.
So I've seen, last year at close to Albertina, there's quite a lot of it. But the one guy from Eskom who's clearing under the Eskom lines came to us wanting to know if this is fangos or whether it's exotic. It was like clearing under the lines every year. And the previous year it's a stretch of about 500 meters, 11 meters wide on a farm just north of Albertina. He said in the previous year they cleaned Port Jackson. There was one or two of these things. So he took me there now, well, not now, this was in May last year. It was 1 year after the clearing. And that whole stretch was like 2 meters high, just this. So and that's what the people at New Year's Veterans sort of tell me that, and so over that where you clear for Jackson or Roykrans.
This comes in, yeah. I mean, certainly
So we now have a proper eradication attempt, because between Harikwa Nature Reserve and Witson, so the whole stretch past Silvia and there's nothing. This is still very clean of it. We only have Roykrans, a little bit of Jackson, and I found 2 populations now of this, and the one is about a hectare of it on a farm very far into a natural, it's just sort of a private nature reserve situation, and it's away from any roadside or anything. And I said to this guy, how on earth did this thing get? He said the only people that was here was woodmakers, guys who came in to cut Roykrans. So they brought it in. And another farmer had the same complaint with Paul Jackson. Then woodmakers came in to cut away ground, but they Paul Jackson.
Is there any, I mean, would it just be a matter of getting funding to, I mean, do you think there's hope for I mean, if there was someone, or was just a very difficult planting?
Yeah, look, that's an interesting question. I think part of it is that, and it sounds silly to say this, that there are actually more important weeds that have winners. And that's what we've got to look at, unfortunately. When we're trying to get funding, you don't really want to back a losing horse. And I'm not saying that there aren't potential. I mean, I think there's another midge actually that makes a much larger gall than this. I've collected it, brought it back, didn't manage to keep it going in quarantine. The scale insect, which has been looked at, potentially one could put more time and more effort. And that's why actually we wanted to base some of the work in Australia, so that we could actually have, instead of, whenever you fiddle with things, as soon as you try and keep an insect going under quarantine conditions, it is difficult. And conditions aren't natural. So that's why we thought, okay, look, let's get some work done in Australia. And unfortunately that didn't materialize. So, but my gut feeling is it's yes, it would be possible, but I think it's better from a funding point of view to get projects going that have more funds. Yeah. Otherwise it just doesn't, it doesn't reflect. We ask for funding because we think, oh, well, we'd like to work on this weed, and yeah, sure, we can work on it, but you as the funder might be a bit disappointed if along the line you don't come up with anything successful.
So I just, as a matter of interest, took a little seed pod, and at the bottom there, that's the match. So it's the size of the tip of my pinky.
Yeah.
And then I counted the seed. So guess how many seeds from a single seed pod? 120.
Yeah, as I said, it would have been hundreds.
And so, they're blown away. They're so small.
I'm not sure that all of them are, so they have quite a lot of aborted seeds within that capsule. But it's that, I mean, and that thing, that as soon as you've got a windy day, which we have plenty of around here, that capsule has these holes, and it just, I mean, they just
it's like
shake.
A shaker.
It's no, they are. It is a terrible plant, and I do wish that we had something more effective, but unfortunately, I don't think at this point.
Yeah, I mean, but I try and do now, if I engage with landowners, is if there's, I mean, so I just found stink clean also, just a few of it now on a farm close to Korikwa. There's a lot of roy grounds in that area. So to say, okay, let's just do these first. Get rid of them while there's still a little of them, so that in 5 years you don't have to fight 3 things. Just get rid of these, and then we go on with the roy grounds. But
yeah, and I would always say that, for people that are doing manual clearing of areas. I mean, obviously, one targets areas that are high sort of biodiversity hotspots, or you might be protecting a special insect or plant or whatever. Focus on those, but also focus on the areas where you've got isolated populations, because much as it might seem more time consuming, you don't want those areas to become worse.
Yeah.
And funny enough, I mean, over the year, I've spent many years working in the Work Nature Reserve, and I know as you go into Potberg, and you get just before where the little stream with all the black wattle is, off to the left, I mean, you've got a golden wattle thick on the right-hand side. But there used to be on the left-hand side a few little isolated. Now that, I mean, and I can remember years and years and years ago saying to the reserve manager at the time, if you have any money, you need to get in and you need to target those few little isolated plants. Well, obviously there wasn't any money, because now it's quite dense, and it just makes it more costly along the line. So I think people tend to sort of see the mass of plants and say, oh, that's where we've got to focus our attentions, rather than actually saying, it's far better to deal with the thinner, sparse. Yes, outside in. And obviously, yes, rivers are important. You need to sort of focus along river courses and things like that. But certainly, I mean, it would always be my suggestion. Try and nip things before they become a problem. Don't wait for them to become a problem, because then as I said earlier, I mean, to try and clear them mechanically, chemically, biologically, it is just a nightmare.
And then just, I mean, be careful with what you move around, because I mean, these both the sites now where we're doing the eradication. When I said this, you just stack it right there, and you don't move anything from there. And we can either get permission from Southern Cape Fire Protection Agency to come and burn it at some point, or you just leave it there and keep the area clean.
We want some more hopeful agents.
Yes.
Keep us positive.
Well, unfortunately, this is now moving to the end of things that are really not so.
Okay, then after this, we'll get after.
So I started with the good stuff. Now we're moving kind of like to the end. So bugweed, and I'm trying to stick to plants that you probably will see around this area rather than countrywide. I'd be here for the next week. But so bugweed, Solanum mauritianum, comes from South America. This again has been quite a difficult project because Solanum's in the potato family. So obviously, host specificity testing, you got to find something that's not going to eat potatoes, not going to eat eggplant. And one of my colleagues who worked on this project, he said it gave him grey hair because he worked for years and years, and he said whatever he brought in, anything that seemed to be promising, go through the host spec testing, it would eat eggplant. So, you know, much as you have, you know,
we really need eggplant.
but I think much as a lot of those farmers are using spraying and all the rest, it was just, you know, they'd just be no-nos. So anyway, we brought in this leaf-sucking bug, Gargaphia, in 1999. So you can see the type of damage. So literally the little bug sucks on the leaf. I think it's usually the undersurface. So it sort of cuts back photosynthesis. Yeah, as I say, been challenging for biocontrol. This insect tends to be a little bit of a boom and bust type of agent where you find it, conditions can be right for it and then it does particularly well. It has never established in the Western Cape. I think it doesn't like the winter here. It tends to be more subtropical, those sort of areas. So it's established in areas of KZN and Mpumalanga, often in the sort of forested areas. So it's doing reasonably well there. And as I say, it is widespread, but in low numbers has these occasional outbreaks. But all in all, not really, and certainly for the Western Cape. I mean, I see plenty of it growing around here. Unfortunately,
we released it.
You've released it, okay.
No.
Yeah, I mean, I tried releasing it around Stellenbosch, didn't establish. And there's also this little flower-feeding weevil for bugweed. So the bugs, the little weevils feed on the open flowers and the actual bud. And then the larvae develop inside the flower bud, and it actually stops fruit formation. But again, established well in KZN, it's established in Mpumalanga. It doesn't seem to do so well inland, does quite well on the coast, perhaps because of those coastal warm winds or something, but never established in the Western Cape, unfortunately. Because yeah, up country it does reasonably well, but it's a bit of a no-go down here.
Yeah, we've also seen it with some of the biocontrol agents that we work with. The plant can adapt to a Mediterranean pine tree, for example, adapt very well in Western Cape conditions, fairly well along the Southern Cape coastline, and then moving with the Natal, we plant Californian and Mexican pines, but then the insect, the attack pine trees, the Cyrex wasp, comes from the Mediterranean. And it moved along, quite happy in the Mediterranean, and Jeff tried and initially said, no, this thing is not going to spread beyond the Mediterranean. Now we've got a Mediterranean insect on a Mexican Californian pine in subtropical Africa. The insect carries a fungus. Both the insect and the fungus are doing very, very well. But its biocontrol agent, which is a parasitic wasp, only survive in the Western Cape and the Mediterranean climate. So the biocontrol agent wouldn't be during the summer rainfall.
Yeah, I mean, I don't think I mentioned, or maybe I did, I can't remember, but with the midge on black wattle, I mean, obviously black wattle occurs in pretty much every province in South Africa. And so when it was doing really well down here in the Western Cape, we were quite excited about the whole prospect of this is really going to be good for black wattle. We got permission from the wattle growers. They allowed us to release up in KZN and Mpumalanga where they're actually growing, providing we had something like a 30-kilometer radius. We had to be away 30 kilometers or 50 kilometers away from the plantations, which I said to them, well, you know, insects don't really know boundaries, you know, and much as we know that it's not a fast disperser, I did say, you know, it could be that along the line it would get into the plantations, but because of the way they harvest, I mean, they're harvesting, I think, every 10 years. So, you know, even if it did get into the plantations, actually, it's not going to have, and in fact, with some of these gall formers, the actual, because it's only affecting seed, not affecting the actual tree itself, so it's not going to affect growth or anything. But sadly, that biocontrol agent has not established well in the summer rainfall regions of the country. And we think it's partly due to the biology of the insect, but as the gall is developing, which it does so here, kind of like in the wet months, the moisture requirements of the gall itself are quite high. So it needs that moisture to keep growing and to allow the insect to develop inside. And obviously you're dealing summer rainfall, it's dry during the period when the insect is growing inside that gall. And we've just had really limited success up in
So this is on pine then?
on black wattle.
Oh, that's black wattle, sorry.
Black wattle, so it's the midge on it.
Okay, this is on the black wattle.
So yeah, so it's established really well, and so we see it everywhere in the Western Cape and the Eastern Cape. But as soon as you move out of the winter rainfall or that sort of, you get areas where you've got bimodal rainfall, into summer rainfall, it's established there, but it's just not really taken off. And we keep wondering, you know, will it ultimately sort of adjust to new conditions? And something I was quite hopeful about doing, a colleague of mine in Australia had actually said, well, they think that midge goes up on different hosts, ones that we don't have here in South Africa. But the same midge, Phacelia rubiformis, actually goes up into the drier regions on these other hosts. And he said it'd be quite interesting to go and survey and collect from that area to see. Anyway, that's somebody else's job along the way. Okay, so that's it in terms of the sort of perennial type trees and weeds. I'm not going to touch really on water weeds for two reasons. I know very little about the water weed project. I haven't been at all involved really, other than to say, and I mean, you're possibly all aware of, I think they now call it Pontidaria instead of Icornia, Myriophyllum, the one in the middle, parrot's feather, Salvinia, and Pistia, water lettuce. All of these have biocontrol agents that are pretty effective on them. And the biocontrol agents, so rather than you going and collecting yourself, the biocontrol agents are available from Rhodes University. And there's an email address there that you can copy down if you want to contact the person that supplies. And when I spoke to Ian about it, he said that they are, usually summer months are the good months. It obviously will incur a courier fee to get the insects down here. I've had some experience working on Salvinia down in the Somerset West area, where we had a local, near my house, there was a little local dam that had a lot of Salvinia on it. We did a release of the weevil that goes onto that, and it seemed to do a bit of damage. There was a bit of dieback, but it never really didn't manage to sort of really get on top of the problem. And again, a lot of the agents are dying out over the winter, so they're not managing to survive through the winter. So one of the approaches that we have, so whilst biocontrol can be, and we always kind of like promote it as being self-sustaining and all the rest, something or approach that does happen is what they call an inundative approach. So in other words, you will just keep doing releases. So each year you chuck some more out. So it's a bit like going and spraying. Instead of spraying, you're just chucking some more insects out. So that's what I would suspect might work best. The biocontrol agents on parrot's feather, I think, work very well. I've seen good success here in the Western Cape. I've just had the experience with Salvinia, where I know it's one of those. I think once you start getting up into the garden roots, it seems to be okay. I think the climate's maybe a little bit warmer. But certainly, as soon as you start going down towards Cape Town, it's just too cold over winter. The insects don't survive. So that's all I have to say about, I think. Let's just check here. Oh, yeah, sorry, no, it's not. So these are just a few weeds that might be of interest to this group of people. And these are weeds really either that have now started becoming more problematic in this area. Lantana is a classic example, and I think just because of the change in climate is now, because this is a tropical weed. It comes from the Caribbean. But we're seeing more and more of it, and it can be little isolated pockets. I mean, I know there's some.
Just curious, is anyone here seeing Lantana? We're seeing it more. I'm just curious. No, I mean, we never used to, but I mean, well, maybe not.
I have one landowner where, when I saw him last year, he had in one field maybe 3 plants, and when I went there again earlier this year, there's like 10, 15.
I'm hearing the same thing. I'm just curious because
Yeah. I mean, it's also why I included it here, because it never used to be something. You know, certainly when I started my career, it just wasn't an issue. Never came down into this part of the world. There might have been, sure, there are some ornamental varieties that people can buy in nurseries, things, but not the really invasive Lantana varieties. And now we're certainly seeing that down here. Unfortunately, because it's also been one of the plants where we've had quite a headache in terms of biocontrol. Mostly because you've got all these various biotypes of Lantana, different varieties that have different color, flower formation. Flower colors be red, orange, pink, yellow, white. The leaf, if you feel the leaf can either be sort of soft and hairy to quite prickly to smooth. They really vary a lot, and because of that, I mean, there have been. Sure, I don't know, if there have been 30 biocontrol agents, they probably have been. I mean, you'll see in the back of that book, if you look at how many agents have been introduced against Lantana. And they might work in certain areas, and then either fizzle out or they keep going, but there's nothing really that's working, particularly for down here. We've struggled a little bit. Having said that, there's a little mite, this area, and you can see it galls the flowers and sort of growth areas of sort of newly growing Lantana. So it actually prevents fruit formation not completely, but obviously it reduces it. I actually brought some with me, which they'll be for releasing tomorrow. So it'd be interesting to see how well it's going to do, whether it's going to establish. I think there's been none released done in this part of the Western Cape. So my colleagues in Pretoria work on it. I think quite a lot of the work has been done in mostly the subtropical areas, so Natal and Mpumalanga. It just so happened that Alan Wood had a lady working for him, and she did a trip to Natal, collected some leaves for some rust spores or fungus spores, and inadvertently had some of these mites on them. And when they put it in their quarantine, well, not their quarantine, but their growing glass house, this thing took off, and as Helen said, they can't get rid of it. So hopefully that happens, and hopefully you have success. So as he said to me, there is a rust fungus on Lantana, but it targets specific biotypes, and they have not managed to get it established on the biotypes down here in the Western Cape. Whereas this mite seems to be less specific. It will go for a range of biotypes. Okay, then, so in terms of weeds which are in the pipeline, and obviously you spoke about, you know, if you were to get funding for a project, where should that funding go? And really for us, you've got to look at, well, where are we furthest along in terms of projects? So there was a project, for example, against Aranda Donax, Spanish reed, where quite a lot of research had been done, and then again our funding was terminated, so it kind of reached a real standstill, and the researcher working on the project subsequently got a job in New Zealand. So interestingly with that, so you're sitting with a plant which has got a lot of knowledge. There'd been a lot of genetic work done on the plant to see where our particular strain of Spanish reed had come from, and from that deduce which are going to be the most suitable agents. So we already have in South Africa, and again, I think it came in inadvertently, it wasn't an intentional release, and I don't know where it came from, but there is this little Tetremesius, there's a stem-galling wasp which is already present in a number of areas, but it's got limited impact. And I know the previous researcher who was working on the project had identified that our strain of Spanish reed comes from this particular area in Spain. The Americans had been working on several agents from that area. And I think the idea is now to try and work closely with the Americans, piggyback onto their project and try and get, I think it's both new strains of stem-galling wasp and also a scale insect, which showed a lot of promise. There was a third agent, and I can't actually remember what it was, but so that's one that's in the pipeline where we've got quite far along with it. So it's not starting the project new, and it's one where we can piggyback on other people's knowledge. They can also piggyback on ours in terms of the genetic work that was done. So, you know, if anyone was going to back something that's a good project to back, it would probably be that one, because you're already cutting quite a lot of cost. And then there's a pathogen that is.
Can I just add something to that, just so everyone's aware on the Spanish reed? So just so everyone knows where that's at, as Fiona said, we're trying to raise, I think the piece of 3 million rand to get the Spanish reed into the country from the work that's being done in America. And because it's been successful there, it's easier to, more likely to be successful here. So we actually, GCBR, Linda and I are working on kind of trying to get proposals out there. If you think about it, all of us work on Spanish reed, it's a very expensive area to control. If we all kind of put in a little bit towards that to get this, it's going to take a while before we can get it in, but the collective reduction in terms of all our operating costs, unfortunately, they're just not, it's difficult to raise the money to get this agent in. So you start to think about it, we're going to try and get a campaign going to kind of raise some of this money, because we want to back the one that we think is going to actually make the biggest difference, and I think this is one of them. So
Yeah, I would say.
if you're interested, please. Unfortunately, we've got to kind of work together and getting these agents into the country, otherwise it's not going to happen. So,
yeah, and then there's also, Alan told me about a pathogen against Tradescantia. People refer to it as wandering Jew. very nice name to call it, but it grows along it sort of grows along the ground. If bits break off, it roots itself.
Is that not root?
No, it's not prickly at all.
Do you know?
Tradescantia will be, it's probably in that green book, or certainly in the new book.
What's the name?
Tradescantia.
T.
TRADESC.
With R there.
Sorry, the text is probably a bit small up there.
Oh.
You see it?
Het groeide ons.
Is it? Yeah, near. Really? Sorry.
So there's a whole lot of different species.
It
So
has a white leaf, like the leaf is white and then dark in the middle.
so he's working on Tradescantia flamensis, but I know there are a whole lot of different species within that genus. So,
And it's promising.
well, he's about to submit a release application.
Exactly.
Yeah.
That's a man.
There we go.
Yeah, I mean, I think again, it's often, if you don't know what it is.
Jonathan, this is.
Sorry, I should have brought a picture of it so you could all see.
I didn't even know.
I just assumed everyone knew it.
No, I mean, I've seen it before, I just didn't know what it was.
I know a different plot as the one during June.
Oh, do you? Okay.
The Melina.
You say cormalina, not chromalina.
Yeah, Comarina, I activated it.
Yeah. That's
Okay.
also got that as a common name.
Yeah.
So,
Well, that's why, I mean, I'm always a bit wary of using it because of that. I mean, you go to Australia and they call black wattle, late green wattle, they have completely different names. It is. Okay, so I think at this point I've reached the end of the actual biocontrol, what agents are available, what they do, how they operate. I can either move into this
We
next
should probably
section.
have promised lunch isn't here yet.
Yeah. So this is really the last section that I'm going to be sort of really doing the talking. That we're going to be sitting and.
Can I just ask, sorry, because actually, can I just ask some of the ones that have been released, we haven't seen effective. Madeira vine,
Yeah.
Pereskia, we've tried to release those needs.
So what did you release? Fenrica, the little beetle?
For the Pereskia.
Two different
It's
ones.
two.
Oh, beetles.
I think they are. They're both Fenrica. You know, again, I think a lot of those weeds are Pereskia also typically a subtropical type weed, and I think our climate down here is just not compatible.
That's what Ian said as well.
I was going to say Ian Patterson's the one really too. So I haven't touched at all on cactus weeds here, which Pereskia would fall under that.
Is that
under that group? And again, the Rhodes group do have biocontrol agents for. I think quite a few of the Apuntia species. I think Apuntia humifusa, Apuntia stricta. There are also, I think, Hypergeococcus for some of the Queen of the Night and things like that. But so anything cactus related, that would go to.
And Madeira vine?
Madeira vine was being worked on by researchers up in Pretoria, and I do know. I know they did some releases up in the sort of Southern Cape, Knysna, and I'm not too sure what became. So there's
We just haven't seen it.
I don't know who you got agents from.
GCBR got some Madeira vine, didn't you? I know
Yes,
you released.
yeah, so we did releases there around November with relay.
With relay.
Yeah.
Yeah. Okay. So she would have got them.
So I don't think it's been.
I suspect again it's just.
That was last year, but I don't know. Also don't know whether it was actually established. I'm not sure. It was first releases.
Yeah. Okay. So
No, it's
it
just
would be
interesting.
interesting to see. So yeah, as I say, I don't, and I can only just assume that a lot of these that are just starting to creep down this way, they're just a little bit out of the climate zone for the insects, but the plants are managing to do quite well.
So do you want to, one thing we could do is we could try and get Alan before.
We can try and get Alan.
Because if we go into this, then I don't know how people feel. They want to, probably.
So look, this is quite, it's relatively short, but it's perhaps an area where a lot of people maybe have more questions. I don't know. So maybe we should try and get Alan.
Okay, and then have lunch and then go into this. I don't know. Thoughts? Anyone? So we can have a quick break while we get Alan on the line. We can get it. And then.
Never say no.
Okay, so okay. The one thing I don't know, I know it's not your thing, but I know nothing about it. But do you understand there's a new pine biocontrol?
Okay, well, I can tell you a little bit about that
Oh, you
project.
can.
So, yeah, I was involved, funny enough, with John Hoffman years ago when we were testing a little seed feeding cone. It used to go for the seeds in the cones of the Pisodis beetle. And then we actually decided to put, so it was specific, I can't remember the species of pine, sorry about that. I think it was, would it be radiata?
It could have been a radiata.
And it was specific pretty much to that, but then it would cause a little bit of damage, but minor damage maybe on some of the other pines. But at the time, we were very concerned that it might be carrying pitch canker, or not carrying it, but helping the ingress of pitch canker, which obviously is a problematic disease of pines. So we took the decision, again, we don't want to get on the wrong side of the pine, pine growers, so let's shelve it. Subsequent to that, there's been huge public pressure, huge, to reopen the pine control story, because a lot of people also felt that maybe we were just being too cautious, that actually what we've got could be successful. I think the one thing one needs to look at with pines is, again, you don't have something that has a seed bank. The seeds are all held in the cones. And I always remember talking to William Bond, botanist at UCT, and he said, well, I don't know why you don't just cut them down, because obviously there's no seed bank. It obviously runs a little bit deeper than that. But yeah, pines are a huge problem up in the mountain areas. So because of that, we've reopened the idea is to try and reopen the project. So there have been discussions with colleagues in Portugal and France. And it's a fellow up at Rhodes University who's now taken up that project. I don't know if he has funding for the project, I'm not too sure, but the idea was to reopen it. So it's not a standstill, but it's ticking under the little bit under the radar. But there is nothing available otherwise for.
There's plenty available. Maybe enough I can give it all.
Well, I was going to say there are plenty of damaging. I was going to say plenty of damaging things, but whether they actually target.
In my previous life, I worked at the institute where we protected pine shrubs and eucalyptus from disease.
I mean, it's an interesting thing because
just pitch canker. I mean.
Yeah, well, as I said, pitch canker
itself. The whole
itself.
peninsula is full of pitch canker. Just to let someone come and inoculate all the trees, but I mean, it's not a self-perpetuating fungus. I mean, actually have to inoculate.
Have to go and inoculate.
It's easier to cut down the tree than do.
Yeah, and I mean, it is amazing how, so there's also this fungus that's been around on the black bottle for a while now.
So, I mean, the pines is the same philosophical problem sort of. I mean, it's the only source of structural timber in this country for short timber. and a large source of paper. At one point, Greenpeace, for example, had on their webpage, they had an article on, against all these exotic trees that drinks all the water and pines and stuff. Lower on the same page, if you scroll down, they had another article saying, I mean, we must stop using plastic and we must use paper bags. Where does paper bags
Where's it
come
going to come from?
So you've got to pick your fights.
Yeah.
And this is, so as much as I hate a pine tree on the mountain top, there's nothing, one thing maybe out there is a telephone line or an electrical line, but it's, but it's difficult things.
Yeah, and I mean, I think it's certainly why, as a group of scientists, the biocontrollers have always tried to not rock the boat as far as industries are concerned. Because, I mean, our experience has certainly been working with people is far better than having them as enemies. Particularly powerful, these big paper, the Sappies and the Mondis and the black wattle.
Those are big international money.
Absolutely.
That is why we work in that money, because it's the only source of money for research in this country.
I mean, it's quite interesting, really, that the wattle industry, despite, and I mean, I do meet regularly with them, they have never funded any of our research, which I think is, I mean, in a way, I think they should.
No, in a sense, I agree. I mean, because especially if you've got seed-reducing agents, and even genetic, I mean, of course, what they're working on is a genetically modified wattle that's not.
That's not, exactly.
So I'll be for.
But you see, that's been a long, I mean, ever since I met with the wattle industry way back, they were working on these clones. I mean, and I still don't know what's become of it. So,
should we take a break and try and see if I get Alan, and then after Alan we can have lunch. Is that good? Good. I mean, obviously carry on the conversation. Hi, how you doing? Yeah, right. So we're just having a quick break. And then basically, kind of went through.
Keep me out of the fields.
Yeah, so I'm also happy to help facilitate some discussion about it. So I think we can do that. Cool. Great. Thanks, Alan. Right. Okay, bye.
Yeah, I just unplugged that. For some reason, I thought my WhatsApps.
Okay, guys, so just before lunch, we really grateful that Alan could come in. So two things. One, if there's specific questions around Fiona gave us a great intro to the pathogen side of it, but there were a couple of questions that maybe you want to refer to Alan. So you can maybe reflect on those and then and ask her to him, because he obviously focused a lot on it. Well, that's his focus. But the second part of it was to talk a little bit about the costing around getting these pathogens released and how we can guide him in terms of what landowners, what we would be willing to contribute, how we can collectively work with landowners to get some of these agents out that needs to benefit. So it's over to you, Alan.
Thanks, Eileen. Good afternoon, everybody. If I can start with, basically, I've got one question, but to give you the background of that. We used to provide hack attack and stump out free of charge to everybody. This was because the costs of producing them were included in the old DFFE NRMP contracts that we had. But those contracts came to an end in end of 2020. We had a little bit of funding in 2022. But like everybody else, we have now accepted that we are never going to get funded again from DFFE. It's certainly not in the short to medium term. I don't know what the long term is happening. Anyway, so everybody's asking and wants hack attack and stump out. To do this, I'm looking at setting up a budget, but to do that, I have to both cost it out, the cost of production, and then also have a good ballpark figure that I can use for income, i.e., how much can we sell the hack attack and stump out? For how much? So the thing is that the direct costs are really cheap. We're talking about potatoes, Marmite, things like that. So that's on a really short term, very little costs there. What does cost is if we're doing on a longer term, we would have to buy things like PDA, which is potato dextrose agar. It's the most common fungal growth medium that we use. We have to buy Petri dishes. We have to get Petri dishes that we use for stump out. We have to get them radiated. And we also have to do maintenance of things like our autoclaves. And then what is actually the most expensive component is that it's to cover at least some of the salary of Gwen Samuels, who is our technician. She's the one who actually spends the time growing these things up, harvesting and sending out. We would also have to include the cost of a courier of getting it to you. We'll probably send everything via courier guy, either to your address or to a pewter box, whatever works. So I'm looking at potentially costing, so for hack attack, costing 10 of the normal little foil packets that we give out at 1,500 rands. That would include the 10 lots of hack attack and the cost of the courier. Costing it like that will cover our first immediate direct costs, but will also help cover, as I say, maintenance of autoclaves. Just to give you an idea, last year we had a quote for doing a service of our autoclaves that was 40,000 rands. So the immediate direct costs are not that much, but if you're looking at the costs around, so that's things like maintenance of our equipment that we have to use, and salary costs, this is why it is more expensive than you might expect. We would look at producing stump out at a similar, but maybe 5 packets instead, 5 of the sachets rather than 10. I'd have to do a more detailed costing out of that. The question is whether you are firstly prepared to pay that, and secondly, via your organizations, whether you can, because I know, for those who are working directly in government, buying things like this can be a bit of a problem. So really what I'm doing here is a little bit of market research of, that's my suggested price. What are your comments about that? Is that acceptable, or is it there's no ways you're going to pay for that, etc.?
And just to help you understand, the 10 sachets, how far does that go? Because I mean,
if you know Hack Attack, the normal little foil packets that we give, that has 0.1 grams of the Coleotrichum spores. You mix that in with a liter. A very rough guesstimate is that's about a hectare. Obviously, there's a lot of stuff on the ground, factors on the ground that will determine exactly how far that goes. The density of the plants, the spray bottles that you use to spray, how much do they actually spray per squirt? Not all spray bottles are equal, etc. So, the size of the trees, how many of the trees that are there that we're going to treat, because one of the ways of using stump out is using Hack Attack is to get it out there that you have some impact. If you're going to treat every single tree, the question rises, well, why don't you actually cut down every single tree? It's an immediate control rather than long term. Biocontrol, it's for Hack Attack, it works where either access is not so easy, or areas that you're not going to get in and hack down. So you get in and you at least get the fungus through those populations from where it can spread naturally, so long as you've got really nice rain seasons. Drought knocks it back. So it's roughly, give or take, as I said, we can't guarantee this, but it's roughly a hectare. So in other words, you can treat 10 hectares from each batch that we send.
So just some feedback from, I know one of the things, we kind of started that conversation with Rudolph, but Jonathan, I'm just wondering, do you ever think that's something that could come out of our business plan from the land care side of it? Would we even be able to kind of factor that in? I don't know how we can cover some of these costs, or be a little bit relevant. We put through herbicide, I think we could put through this kind of thing.
I don't like speaking on behalf of you, certainly it's moving.
Yeah.
Cost to specialist people, these kind of offices, cost for PPE, for other sites, and agriculture is key on the biocontrol thing, so I don't think to better cost.
You see that?
Just looking at it's 1,500 for 10 hectares, it's about 150 per hectare. I think that's going to be to see.
Yeah, that was my gut feel. You obviously still need a team though to go in and apply it.
You still need to.
But it's not as intensive in terms of having to do the whole. I don't know, other
It sounds fair, but we have to talk to the people.
above you. Yeah, I understand. I was hoping that Linda and Ruby would be here, but they're traveling the world. Yeah.
I'm going to speak from the point of view of a volunteer because I don't do this in my professional capacity. We clear hack here as volunteers. So from a volunteer point of view and having applied and having used this product, having used Habitat, I would say that the 1.5 is reasonable, especially for those 10 foil packets. We work in areas where it's fairly dense, so it does work quite well, especially up in the mountains where you get a lot of humidity. And I would also say, because we also work with industry partners, we try and involve ladies with cheese to get funding and stuff like that. I would say for them, this wouldn't be an outrageous cost. So I think they would be willing to pay that for the effect that it is going to have. So my answer is yeah, I think it's a good deal.
Yeah, great, that's good. And then of course, always difficult to ask Cape Nature. Because I know you guys probably don't have control over these budgets, but do you think, I mean, what do you think if you were to talk to your superiors and ask them or you yourselves, is that something that Cape Nature could rub together?
Like usually also we have to talk to the people high up and discuss it with them.
Yeah.
I mean, also think we want to sit down and do the cost analysis. What would it cost and how many times to go back and
Cape Nature should go to GCPR and other non-governmental supply the funding and they will supply the labor.
No, that's up to the individual organization, because the Cape Nature used to fund us. That's how it used to work. but I agree. I mean, there's partnerships that you can. I think one of the challenges within government is some things go through the procurement process, and then it's very difficult, even if the funds are there, to actually access some of the funding, even if they have it. But partnerships definitely are part of that. Sugarbird, I don't know, you guys.
I don't think it will work on our project because we just cut. So most of the areas where if we were to provide the gum, that's the most part of the trees which you are dealing with. You don't have the control on it. So if there is a certain one which we can get from there, then we can.
So you're saying in the areas where you work, there isn't a lot of hack here?
No.
Okay. Okay. So it's not necessarily applicable in where you're working. Yeah. So you haven't got one for eucalyptus kind of in the wings there, have you, Alan, for us to release something like that? No, we're just I'm pushing my luck there. I don't know, any other comments on the cost? You froze. I thought he was just being very still. Alan, are you still there?
Yeah, I'm still here.
Oh, it froze for a second. You were completely frozen, so we thought maybe we lost you. But you are still there.
Yeah.
It is recording. I hope it is recording. I hope it is.
What about, there was a question earlier from somebody about whether hack attack actually works on any of the other hackers besides Serisia.
No, it's only Hackia sericea. It's not gibosa or any of the others.
And so that was one other question. And another one was are there other pathogens that, I mean, it sounds like the stink bean one kind of is on its own. Are there other ones that need assistance in terms of on releases?
No. So of the pathogens that are active in the Western Cape, there's only the Port Jackson rust and then the stink bean rust, and they do their own thing. Then it's hack attack and stump out. Those are the pathogens we use down here. I've got one pathogen in the pipeline that might be of use I think that will be of interest in the Southern Cape. This is a smut fungus on Tradescantia, the impolite old name is Wandering Jew. So nowadays people are calling it Wandering Trad, but I'm not sure if people know it. It's a weed of forest areas. So it is very limited in its distribution, but obviously our forests are small but incredibly rich. So it's not a widespread weed, but it can have a very high impact. KZN,
So we will keep Googling it.
So basically, I've just put in for application to release. So hopefully within a year, I'll be getting that out there. But if people are working in forest areas, to be aware of the weed, and if you've got a problem, the pathogen is on its way. Yeah.
So we definitely we were Googling it and looking at pictures, and we all went, well, we work in forest, so we all saw it and went, oh my word, is that what it is? So if you need a place to release it, we have suggestions.
Yeah. I mean, I'm going to be hopefully releasing it throughout its range. Yeah. Again, scraping together money.
Yeah.
Funding is an issue, as you've heard from Fiona, I'm sure. But anyway, yeah, we will do what we can. Yeah. Okay, so, but yeah, that's basically it from a pathogen side. There is the rust on lantana, but very few types of lantana are susceptible. And as far as I can tell, none in the Southern and Western Cape. Yeah, and that's basically it. All the other agents in the Western Cape is Fiona's insects.
Good. Other questions and I hope that feedback on the costing is useful for you, because it sounds like most people in the room feel like it's those that have Hackia feel like it's reasonable. We can also share.
Yeah.
Maybe just what would be the costing on stump out?
Oh yeah, sorry, that's a good question, Stump Out.
This is a bit of a thumb suck. We're going to have to look at how much it costs per batch. Again, the direct inputs are not so much, but it is time, because stump out takes a bit over a month to produce. Most of which it's just sitting in a production lab we have to grow it up at 18 degrees with modified Petri dishes. But per batch we put in, we usually get out around about 300 milliliters of the oil, which then makes about 20-30 sachets. So I'm guessing. So one of the things is, if we supply just a few, then the direct costs go up per item. It makes more financial sense to supply a set number of sachets. So I'm guessing it will be in the order of 5 or 6 sachets, each one of which you mix up with 400 mils of cooking oil. Again, how far that goes is all dependent on how you use them. So the density of trees, the size of the trees that you're cutting down, so it's, again, very difficult to estimate how far it goes. But I would say it would be roughly the same price, so about at 1,500, which would include courier, and that would be for, I'm guessing, 5 or 6 sachets.
How many sachets So, and we're just trying to compare, we just looked at the area where we applied it in our area. And we just wanted to estimate how many sachets did we use there? Remember?
Yeah, we use both the 1 kg jar and the sachet.
Okay, because we were trialing the two different things. Okay. Yeah. So we checked it the other day and it looks like it's been effective there.
That's good.
So, and does it depend on the, because I know when we checked that area, there were two different types. So the costs change depending on the different way it's mixed.
Yeah, so I mean, what I'm talking about supplying is the stump out that we've supplied all these years, which are the spores mixed in in mineral oil. What we trialed was doing just the hyphae. I know because I looked at this that it does not have the same shelf life as the spores. It would have to be used immediately after production, the hyphae. But but you know, as I said, so Eileen, that's the thing. There's the trials that we've done with you to see whether that works. So certainly at the moment, we wouldn't be supplying that for use. We'll only be supplying the old way, which are the spores in the little sachets.
Once you've tested, yeah, okay.
Yeah, those that were tested a long time ago and still working. Yeah.
Does that kind of answer the question? Any other questions while you've got Alan on the line on pathogens or anything that?
You have anything for bramble?
Bramble. Oh,
yes. American bramble.
So we've got a long list of things we want you to work on, Alan. I know you're also looking at retiring at some point, but
well, bramble is interesting because there has been a desire to work on bramble for a very long time. But the taxonomic of what we actually have and what we call bramble is very complex. There are many different species that are just termed American bramble, but they're not actually. Yeah, there are a number of papers which have brought a bit of order to what's going on. There are some very good pathogens available. There are also quite a few insects. It's getting funding for this. The plantation industry should really be funding because bramble is a massive problem in the pine plantations, etc., up in KZN and Mpumalanga. So, I mean, yes, there's potential for bramble, but whether it actually is American bramble that you have and is a problem in the area is something that needs to be verified.
Any other? Sorry.
What about Peterson's curse?
Patterson's curse. So that was one that is that what you mentioned?
Yeah, I mean, there are, and I don't know about the pathogens, but Australia has got a very good biocontrol program against Patterson's curse, which we could potentially piggyback on. So for us, it would be a brand new project, so there would be big costs involved, but our costs would be cut down a little bit because there are already insects that have been utilised, earmarked. So there would be some testing we'd have to do with it. There was a project we actually put forward to GFFE in our last tender. So it was going to go forward, but obviously now.
I know there's one that I don't know whether Ian mentioned it to you around that yellow iris.
Yellow iris.
So that yellow iris, apparently they have got a biological control, but it's sitting in quarantine. Yeah. The last hurdle, and they ran out of money.
Probably another year of testing that needs to be done.
And that's all that research is now just sitting there in quarantine. So unfortunately, these things need funding, and unless we raise the funding, or motivate or advocate, it's going to be hard to get more legends out of it.
just on that, you can guesstimate that it's approximately R1 million a year that we need to test. So that pays for a senior researcher, a technician or two, going to Australia to collect the things. That's really expensive at the moment with the exchange rate. But once you've got the potential agents established in quarantine, it really is, it's paying salaries. And obviously overheads, electricity. Well, the quarantine glass houses are very expensive to run in terms of electricity. But that's really where the costs are. It's salary time, and some basic overheads, really. So yeah, anyone come up with that, we will gladly do Patterson's Curse for you. It's a project that's waiting. And actually has been waiting for quite a few years. We should have got involved in this a long time ago, Patterson's Curse. But yeah, that's, you know, if you think how much money is spent on clearing,
absolutely,
versus the development of an agent, which is, it's no silver bullet, but over years it adds to, you know, the overall. So in over the long term, the cost benefit ratio is actually very, very good. It's just convincing the politicians to put the money where it needs to be. So your help is gladly appreciated for putting pressure on the politicians.
So yeah, and we are going to try, I think we are starting with Spanish Read and try and put pressure, but in that process, trying to put pressure in general to raise the profile of biological control. That's kind of where we're going. So please speak to your organizations to try and kind of raise a profile, because we don't, then it's not going to happen. Thanks, Alan. We about to have lunch. I'm sorry you can't join us for lunch. We could courier you some lasagna, but yeah.
Everybody loves lasagna.
Thanks very much, Alan.
Okay, thanks, Eileen. Bye.
Right, bye.
Bye.
You all guys had a good lunch? All good? Full bellies? Now I see the eyes are going to start drooping.
I was going to say, now's the time everybody's going to be falling asleep.
Yeah.
Anyway, look, this part is as long as short as you. It's relatively quick, because in terms of implementation of biocontrol, if I get rid of this thing now. There we go. It's often easier to show people how to collect by actually doing it, rather than to show you pictures of things. But I think I can at least give you an idea, and also a bit of the information regarding what insects need to be implemented. I've tried to touch on it during the course of the presentations, but there are more specifics here that I'll go into. So obviously, as a first point, not all biocontrol agents need to be implemented, and some disperse perfectly well on their own. I've touched on those in the morning. I think one of the important things is, and particularly when you're starting out, trying to get nursery sites going, it's really important that your release sites are, you think about them quite carefully. If you're going to be going back to doing recollecting from that site, don't make it in an inaccessible area, and don't put them in an area that's going to be targeted for clearing or burning 2 to 5 years down the line. You've got to think of these biocontrol sites as long-term opportunities. I think very importantly, get permission of the land before you proceed. I've been often on the receiving end of very irate landowners, and I've always had this sort of attitude: it's easier to plead, forgiveness than ask permission, but it's not always that way. So I think it's very important to get permission. Then in terms of how you get hold of biocontrol agents, well, in the past it's been relatively easy, and as I said earlier, it's been a free thing. You could get hold of, in this province, it was getting hold of Relay. Everybody knows her, through Working for Water, which was DFFE. She would go and collect the insects, or teams of people would collect the insects, and then they was getting sent out to the public. And obviously the other way is receive training and learn how to do it yourself, which is kind of like now the point of this whole session for you guys. And then obviously make sure that where you release the insects, you check for establishment, because you know one of the things that DFFE used to say to us is that they want every single release point. And yes, it's very important to have. So they had this big, I think it's on some WIMS setup, which isn't accessible to everybody. They wanted to know where all the release sites are. Well, a release site's all fine and well, but you might not have establishment. So it's meaningless. That little GPS point that they've got hundreds and hundreds of GPS points of releases that have been done over the years, it's meaningless. What is meaningful is, are the agents established at that site? So it's far more important than where you've actually released them. So it's always important to follow up. Where you've done a release, has it established? Might have spread slightly, but those are really, really important issues. And then once you've got that agent population, so obviously with an initial release, it's important to keep those sites where you've got a good population established, keep it as what we call a nursery site. I spoke about them earlier. Those sites become really critical for going back to doing collecting and redistributing insect or pathogen agents elsewhere. As I said earlier, mass rearing is a completely different thing, and I'm not going to go into it at all here. We don't do any mass rearing for any of the agents on trees. They tend to use it for the cacti and the waterweeds. But we don't. So it's all field collection, and we rely very heavily on nursery sites, or we rely on sites where we know populations are reasonably good. Okay, so in terms of some collecting methods, because again, it's not the same for all species. Depends what sort of insects you're working with, what life stage you're going to collect. Are you going to collect the insects themselves, or are you going to collect galls or larvae? Are you going to collect adults? So usually with the gall wasps and the gall midges, the easiest thing is to go and collect the galls. So you would harvest galls at the right time of the year, and then you can redistribute them. In terms of I might broach, I think I broached it, so I'm just going to hang 10 for a minute. For the seed weevils, so that can be the acacia weevils, Hackia, Sesbania, we do what we call shake sampling. So we have a beating sheet, which is normally a heavy duty plastic. It's better to use a light color, so a white or a cream. If you use a black sheet, you're not going to see the insects because they're going to fall and they get lost in the black sheet. So it's always good. A classic thing with many, and I said, you've got the droppers and the clingers. So those insects that drop, classically an insect will behave dead. So it's a normal behavior. So you might look on the beating sheet and think, oh, everything's dead. They're all lying with their legs all pulled up, and give them 2-3 minutes and then they start wandering around. And then we use something called a pooter or an aspirator. I don't know if you're familiar with them or not. Many people I've trained before have no idea what they are. I will go into it a bit later. Yeah, and it's the same method again for the Hackia, the little bud weevil. Shake and things drop onto a mat. And then there's hand picking, which I spoke about. So for example, the Sesbania, the big stem boring weevil on Sesbania, you use hand picking. So you go, you stand by the plant, look at it, and literally, and obviously some insects are really tough. So that big weevil on Sesbania is quite a hard insect. It can take quite a lot of handling. Others don't really like being handled too much. So again, even using a pooter, there are some tricks that you do. Put a little bit of tissue paper in the bottom of your pooter, Because for an insect, being sucked into a container, it's like massive force. It'd be like putting you on under massive G forces. So you're being sucked into this thing, flock onto the bottom of the plastic container. So you want to be gentle. You want to put something in there just to break their fall a little bit. Your insects are going to survive longer. You're not going to kill them even before you've finished collecting. Okay, and then just in terms of application of pathogen spores, there are at the back some brochures, and I'm not sure if all of you have seen them, but please have a look. You're welcome to help yourself to any of those paper brochures that are there. I know specifically there are two, one for the application of hack attack and the other for the application of stump out. And the whole method, the process is all explained on the back of the sheet. I think even I haven't had a read of it, but I think they even explain. So this sort of tool that generally what you do with hack attack is you have to go and you wound the stem of the plant. So you need something that's going to cause a bit of damage and get through that outer layer of bark. So generally people have used broomsticks with nails and anything that's got some of these sharp little prongs on the end. You can literally just walk around, depending on the size of the tree, give it a couple of wounds around the perimeter, and then you would just spray, using a spray bottle, spray the fungus onto that wound and leave it. And then I think stump out, and I've never applied it, but I think you use a paintbrush and just paint the stumps. But all of that information is quite well laid out in the on the brochures there. There are also hack-year manuals. I see quite a few you've picked them up already. It's quite a good little manual that's come out for management.
This is our onboarding tool for when we do architects.
Yeah, okay, so that works great. The only thing is if you've got a really big tree, then you're going to get, so it's, I don't know about you, but I don't really like getting sort of steered by those things. So I mean, we
also work nice.
Anything that's going to wound, and often it's useful to have something with a slightly longer handle, because those spines are horrible, actually. I mean, then you get these like little sores that, so it's just a horrible plant generally. But yeah, that's the whole idea of causing a wound, whether it's a slash with something like that, anything that the fungus can actually get in through. Okay, on to pooters. So a lot of people say, oh, well, how do you make a pooter? And in the past, again, it's something we used to supply pooters to people. And I then got very tired of making them, and I said, well, they're very easy to make. You need to make your own.
There's the one guy in George that used to work with Relay.
Yeah, so that Herson, and I think I've spoken to a number of people. So Herson actually started off he was one of the contractors on one of our biocontrol implementation teams. So I did training with him years ago, and he's a brilliant guy. He's very passionate about what he does. He just got it. And his team, it's really interesting how these implementation teams, that team up in George, they were a really cohesive team. They did brilliant collecting, and he was a really good leader. And I know he's recently made some, and I'm not sure.
Yeah, so
was it for you guys?
Us, yeah.
Okay, I know he'll charge you for it because he's.
So we paid for it, but the thing is, for me to get the right tubes and the right glue mix and stuff that's not toxic and stuff like that. So he's got all of that readily access. So it's just.
Yeah, I mean, it is really a fairly simple process. And look, the main thing is actually, so you can buy that flexible tube, be it from builders, be it from an aquarium shop. And pooters come in, as you know, they come in different sizes, depending. I mean, for those little midges, we've got tiny little pooters where we're using aquarium tubing. And what works really well is, in order to sort of get that tubing through, so you make the hole on the lid slightly smaller than the width of your tubing, and then just heat the tubing up. So you can actually force it through. Because I know a lot of them, I mean, I've got a puta's just got a whole lot of crackies putty or something just to keep it becomes quite heavy. But for the smaller putas, it's very easy just to heat the tubing up, force it through the lid. The critical thing is one side, you need to have like a little bit of mesh. Yeah. And that's the side that you suck on. For the pure reason that the other tube is what you're using to suck up the insects. So you don't want, and God forbid if you suck the wrong tube, because you're going to get everything that's in that jar in your mouth. And it happens so often where you're busy in the field, you're not thinking, you just grab your puta because you're not really looking, suck on the wrong end, and then it's a mouthful of insects. So generally speaking, I will often wrap something around the side that I know I need to suck on. So it'll be like a bit of tape or something. So I know when I grab my puta, because otherwise some people use different color plastic, whatever, but the side that you're sucking on is protected by a little bit of mesh, so you don't get the insects coming up into your mouth. You will get bits of dust and stuff, but that's manageable. Yeah, and I think that's pretty much it. So literally you suck on the one end and in the insects come from the other end. And it's, as I say, important to make your putas according to the size of the insect that you're. So those big insects that cling, a puta's not going to work. You're going to suck until you're blue in the face. You won't manage to get them. But small insects, yeah, it's easy. So anything that falls onto your beating mat, or even if you see things on the plant, you can just take your little tube, suck, and then into the tube it goes. So it's, and then normally what we do is once, okay, these are really just to show you the process. And I've actually got a beating mat with me. We're not going to find anything at this time of year, unfortunately. It's a very bad time of year. But that was doing some collecting on Hakia. What is quite useful to have, besides your beating sheet, is a pole. So that you're not having to hold on to a prickly branch whilst you're shaking. You can just give that tree a good clap. It's really good for letting off steam. And a pole with a hook on it is also quite useful, because sometimes you go out and you can see, oh, those branches right up there look really good. They've got lots of pods on them. Chances are they're going to have lots of insects. You might want to get your hook over the branch and give that a good shake so that the insects will fall to the ground. And again, so similar principles where, and as you can see, you shake a wattle tree, or shake anything, and you're going to get all sorts coming down. You get spiders, you get snakes, you get everything falls out of the tree. And it's then a case of looking quite carefully to suck up the insects. Then, in terms of packing those insects, so with your puta, you can keep sucking away till you've got 1,000 insects, but it's actually not good to keep that puta too full of insects at any one time. I mean, often you're out in the field, it's very hot. So usually I just do a mental count, and depending, so these storage jars, I use honey jars that have I've cut holes in the lids, they've got a bit of gauze on the top. And I always put inside a little bit of tissue paper, sometimes a pod or two. They can, it gives them something to cling on to. So normally I'll do a mental count in my head whilst I'm sucking up the insects. So I know those jars will take up to about 100 insects. And that's quite a good number. You don't want to overload them. You want to put 1,000 insects in there and then they sit for a week. You're going to end up with dead insects along the line.
Yeah, I had a problem with Bavaria fungus in one of my.
And that's the other thing. I mean, I know that and the weevils, my first few trips over to Australia, I can remember coming back and I'd collected, I think, 10,000 melanteries. So I had lots of them. Very pleased with myself, get them into quarantine, and I was putting them into quite large sort of those bread container, bread saver things. Quite large numbers, and the whole population got wiped out. because of Bavaria. So yeah, it's better to have fewer in more containers than lots in.
Bavaria is just like a white mold, it's a fungus that kills the insects. So
and it spreads through a
very
population
quickly,
incredibly
especially
quickly.
if there's too much moisture.
Well, that's the other thing when there's
So if
moisture.
there's no fungus, how long can they stay like that if there's not too many? And how long before you release?
Well, it depends how you keep them. So for example, collecting in Australia, I've often been there for 3 weeks at a time. The critical thing then is, if you've collected, and that's another reason why we use the tissue paper, is actually to absorb condensation, because you get quite a lot of condensation building up on these containers. So if I'm traveling, I don't often put a pod in there. I'll just put the tissue paper. They're always kept in a cooler box. So when I'm out in the field, I've got a cooler box with ice bricks. They go straight into that, so it's cool. And then overnight, I keep them in a fridge. And then you can keep them going for quite some time if you keep them cool. So around sort of 5 degrees or around that sort of temperature. You can keep them going, not months and months, but you can keep them going weeks.
Yeah, I mean, 2 weeks or so maximum, I would say. But the other important thing is that the lid of the ones that you keep them in also have pores so that it ventilates.
It ventilates. Yeah.
So, and then, yeah, it's just to keep them separated.
Yes.
Smaller amounts is better than.
Yeah, I mean, normally if I'm traveling, I might only have 50 in these containers. It's when I'm shipping them. So if I get back to South Africa, people are wanting insects, and I've said to people I relay, when you collect and send out insects, don't put more than 100 in there. Otherwise, it's too many. And especially if they sit for 5 days with a courier, it's hot. Much as you say to a courier, please keep everything cool. They don't, and they'll leave the box sitting in the sun.
So if you work with insects like that, always keep your cooler box in the shade.
Yeah.
Never leave it in the car in the sun, even if it's in the boot. It becomes very hot.
Yeah.
Take it out and put it under the car in the shade, but always in the shade. And if you've got blue ice blocks, put that in, but wrap that in tissue paper or a towel or something that you don't have to.
Yes.
On that.
Yeah, because immediately we've had that too, people putting ice blocks and then you get a lot of condensation building up and then the insects drown in it. There's so much condensation that builds up in those containers. That tissue paper becomes drenched, you get a layer of water on the bottom and literally the insects will drown.
So the other thing is how much is too much actually in terms of per job?
So, well, for me, anything more than 100. If you're going to put more than 100 melanterius in a little, and I have some in the car, so I can show you the size. So this is slightly smaller than a standard honey jar. It's not standard honey jar size, but I wouldn't put more than 100 in there. And again, depends on the size of it. If the weevil's bigger, I would put less. If you're dealing with transporting the Sesbania weevil, those big Sesbania stem boring weevils, I probably wouldn't even put them into a jar. I'd leave them in a gauze bag or a pillowcase, pack them carefully, obviously, in a box that they're for transport. But you've got to be really, you just don't want too many. And actually, it's the issue with fungus is the big issue.
Yeah. So also, once you've used them and emptied them, wash them with soap water, rinse them properly, and let them dry properly before you use them.
Fiona, with that number, just out of curiosity, how long would it take? I mean, is that a day's work for a team to collect that many? Is that you on your own doing it? I mean, I know it's variable depending on how many there are, but just to give an idea, how long does it take?
It's really, as we like saying, how long is a piece of string?
Yeah,
it's very difficult to, interestingly, the melanterius weevils tend to aggregate. So you will often find whether they're overwintering, and I've collected weevils for foreigners who need weevils at odd times of the year. under bark, and so during the winter months, these weevils actually go into like a semi-dark. It's not really a dark horse, but they overwinter, and they get into little cracks and crevices under the bark, and that is really low. I mean, to collect that amount of insects can take a long time. To collect 1,000 insects takes a long time because you're literally digging under little bark, peeling off bark off trees. Trying to find little groups of 3 or 4 weevils. Sometimes you can hit a mother load and you can hit 20 weevils all sitting in a container. One thing the weevils do quite like, and particularly the ones on Port Jackson, they use the old Uromycladium gall as a refuge. So they will often overwinter in the old gall, but it's always the old. If there's openings, holes and things in that gall, they like that. But if it's, I mean, I've been to sites where literally you put out your beating sheet and you tap the tree and they rain down, and it can be half an hour or it can be 3 days. It depends.
I mean, we were 2 teams with the Roy Krantz, of 3 or 4 people. It's just easier if you're about 3 people at least to work with a sheet. And stuff like that. And we collected, but I mean, it was also training, so it was not very efficient, but I think 10 jobs, 1,000 people in a day. Of course, they weren't very dense population, so you actually had to hit a couple of trees to get 100. So.
I mean, it's difficult to say time, and I know initially when we first got the teams out working in field, obviously they were paid per day, and we didn't really have any targets for them because we weren't too sure how well they would do. Well, actually, the team in Probo, they were quite lazy, so they just thought, okay, well, we're being employed for the day. Doesn't matter if we collect 5 weevils or 500. Whereas the George team, they were really amped for what they were doing, and they would collect thousands. So then we started saying, well, maybe you need to collect 1,000 weevils a day. We tried to put numbers on it so that, but then what happened, they would sort of collect their 1,000 weevils and there'd be tools down. Okay, we finished for the day. So it's always a difficult one. And I think really that's where, you've got to have a good contractor who manages his or her team. And you can see, sometimes it's hard work collecting. And it's always good. I always say to people, you want insects, go and collect them yourself, and then you really appreciate what you do have.
I mean, at first when Relay just sent us insects, I mean, you sort of get us to the insects, and they can stay with the car. I mean, collect them yourself. You treat them like baddies.
Yeah, and because you know the effort that's gone into it, so you.
And when you shake and you get a sheet, yes. Do you have to worry about not getting, or are there other weevils that could be that you need to actually be able to identify the specific one, or is it like?
I think here, I mean, when you're collecting here in South Africa, I don't think.
You're not gonna.
I mean, yes, you might. The other weevils that we get here generally are quite a lot bigger.
So they look different.
They look very different.
Okay.
In Australia, it's a slightly different thing. I mean, and you are getting, it's not just the Melanterius. There are other species that will fall on. Some of them look slightly different. Some are quite similar. Here, I think you're.
Okay.
So, I mean, there's always this process where you're packing up, or you're sucking up, putting into, and then at night time you literally sit and you sort of empty everything out, and you're going through the whole process again to do this sort of pre-screening, pre-sending screening to make sure that you. But I think that's not. I mean, it's a good question, but I think there's not. I mean, yes, sure, we might be taking a weevil from here up the coast a little bit, but I don't know that it's a big issue. Okay. So then the next thing often, and it's a question we always asked, okay, well, when is the best time to monitor and when is the best time to collect? And again, this is something that varies from insect to insect, pathogens, whatever, and it varies from plant to plant. I have here, and there's probably enough to give everybody, this little page here was taken out of a book that we got, a little booklet that we got together, actually hoping that at the time DFFE would fund it. So this only reached its draft stage. It was never, we were hoping they would be able to publish it. They published all sorts of things. They came out with playing cards, and they spent a lot of money on other things. And actually, when we saw them, it was embarrassing because they were, and I shouldn't speak ill of them, but there were a lot of errors in stuff that they'd published. And I thought, why did they spend all that money publishing something that's incorrect? But anyway, they didn't fund this. It does need to be updated. So not all the agents that I've spoken about today are in here, but you're all welcome to have one, and I think there's about 20 there. But where it's really useful is that there's not a lot of words on there. And I mean, I was saying to Eileen, in my experience working with guys in the field, they don't want to be reading through screeds and screeds of you need to do this and you need to do that. So it's a very quick, you can have a quick glance. And for example, the monitoring times are in yellow, the collecting times are in green. And these are the best times for doing that. Obviously, there can be a little bit of, leeway in between. So it's a really easy thing at a glance. You've got the months across the bottom, and it's very clear. You can see, for example, the black wattle, midge, June, July, a good time to collect it. But all those other months, March, April, May, June, July, August, September, you can do monitoring. You can see whether it's present or absent. So it's a very useful little pocket field guide. Again, if there's any funding floating around, it does need to be updated. It would be nice to be able to print something that's a little bit more sort of substantial than this little booklet, but it works fine. And yeah, I think it's quite a useful thing to carry around. There is also it's kind of like a Biocontrol app that you can have on your phone. So if you're like me and you use an iPhone, unfortunately you can't use it. It's not for iPhone users, so it only works on the Android system. But what is really nice, again, if you're in the field, you can just click black wattle. Yes, this agent, so you can say, right, Dacinura, the gore midge is present, Melanteria is present, or absent, whatever the case may be. And all that information also then gets sent off to an Excel spreadsheet. So instead of having to take a little notebook or try and remember it, we all generally carry our phones with us. So it becomes quite a nice thing just to capture quickly. I have a contact at DFFE who's prepared to give a training course on how to use the app. It is relatively straightforward, but I don't know if it's something that this group would be interested in, but I've chatted to you about it, and I think it's a useful tool. I mean, a lot of people prefer to use iNaturalist or some of these other, but this is specifically geared up for biocontrol monitoring rather than weed monitoring. So it's really nice if you're wanting to capture presence or absence of biocontrol agents. So again, for the guys that are out in the field, they're going to be doing, say if you've got teams of people, you want to get out there and you want them to go and collect Melanterius for Acacia Mearnsii. So, a lot of, if I was asked right now, well, go and collect, I really wouldn't have a clue where to start because I haven't been out in the field for a long time. And it's really important to have feet on the ground, actually, making sure, okay, well, they know Melanterius is at that place, it's quite abundant there. When we had Herson and his team up in George, they were on the ball, they knew where the stuff, I could say, I need 1,000 Melanterius, he knew where to go. And I was chatting to him last week, and he said, unfortunately now, because they haven't been funded for the last 3-4 years, you get out of that. So suddenly you're not out in the field anymore, you're not doing the monitoring, and that knowledge is lost. So it all has to be built up again. And it's really useful it is useful information.
And the landscape changes.
The landscape changes, yeah, things get burned, cleared, whatever
Yes,
the
I mean,
case may
so
be.
Relay contacted me, for example, for biocontrol release, she had available for Jackson. But I don't have too much for Jackson in the area, so I went on to Cape Farm Mapper and the layer for Jackson, showing me nice patches of that. I went to those patches, not a single one had more than like 5 trees on, and they were like dense stands apparently 10 years ago. So
Yeah, that Cape Farm Mapper app, hilarious.
yeah, so
There's somebody set that all the time. Really.
Tell them, they need on the ground stuff.
Yeah, that's the.
yeah, so I don't know where they get the alien invasive information from. The other stuff is fine on that, but the alien invasive stuff on there is really. But I mean, the point is, it might have been accurate 10 years ago. But it's
not any longer. Yeah, and again, I think this is where it is really good to have people out there on the ground, teams of people that, you know, they know they're passing these, you know, patches of weed all the time, and they're marketing all the time. They're marking. They know it's there. They know the agents are there. Or oh shucks, that place is gone, so you kind of write that off. But if you're not on the ball, you lose that. So then it becomes this whole process of having to source where the insects are again, and it's that again takes time. So I mean, I think this is what I was saying to you a while back, Ellen, is that, you know, much as I know there are people really keen to get involved with, say, collecting melanterious insects, but they don't realise that, well, it's not a case of just pointing them, go there, or it's in your backyard, you can go with your beating sheet. You might have to travel for 50 to 100 kilometres before you find a site where they are. And then travel again, because you couldn't get as many as you need from. So it does take time. And this is where consistency within teams and having those teams that go out all the time, the people in the field, I mean, it's invaluable. It's very, very important.
We just, almost all the trees.
Yeah, I mean, I think what's interesting now, not that long ago, I actually drove back from Eastern Cape, and as soon as you get into the Western Cape, it is pretty much everywhere. It's but it's really what is critical is that very small window of opportunity for collecting, and it's the same for the melanterious, the adult weevils. Sure, there are other things. You can collect seed pods with larvae and try and moving, but it's not such a successful way of doing it. Best is to get the adults, and you've got a 2 to 3 week window for doing that collecting, and then they are gone. Well, they're there, but they are very difficult to find, and then it's just going to become hard work.
You say that the ones that are need the most work are the newer ones. I mean, because I guess what's interesting is we all say that they're everywhere. So, which ones should you think we should, focus on?
What, in terms of everything that I've spoken about today?
Well, these ones.
Okay, well, these are both very widespread agents, both that you're up on the screen now. But the melanterious weevil on Port Jackson is that could do with spreading. I think it's not everywhere, and it's actually been quite a successful agent.
But it hasn't been spread
It hasn't been spread so widely. I think also maybe because people feel, well, they've got the fungus. But something that we've seen, when any tree is put under pressure, so obviously it's under immense pressure from the fungus, they actually start pushing out pods because it's like a survival mechanism. They realize I'm dying, I better start producing seeds. And very often they will produce quite a lot of seeds in that last stage of their life. So it's quite good, and the two agents work very well together. And you can have the fungus and then the seed weevils there to kind of mop up the seeds. So that's one that I would say still needs redistribution. Again, on black wattle, it isn't everywhere. I mean, in this province, yes, it's been quite widely collected, but the midge has kind of put pay to. But you can still collect if someone wanted for example, melanteris for black wattle, I would advise them to go and collect it off golden wattle. I mean, I know at Duhoek, for example, I've done collecting there, where, you know, because the trees are still producing, despite having the gall wasp there. There are years where it produces, they still produce a fair number of pods, and that's what the weevils will now, they've moved on to there. And they, you know, so you've got to search around a little bit, but you've got options. You can collect that same species of golden wattle, silver wattle, and there are still sites where you can do that. Any of the others? Yeah, obviously then there's the two new agents. There's the datanura so the gall midge and the gall wasp for silver, that group of wattles. But really, as I say, at this point in time, it's still a little bit early to have people en masse coming. And so I think, you know, for maybe the next year, 2 years, it's really going to be up to us as researchers to try and identify some good nursery sites. We've got a site, I sent some up to Albertinia, for example, that area, they've got silver water, I know it's established there. So, you know, it's nice to have that feedback, and it's good to have those nursery sites that are in progress. And I think that's how I see the next few years. And then obviously it can be rolled out to the relays, who will go and, you know, just collect in large numbers. But I think at the moment it's a bit early. Okay, and I mean, I think, again, we've touched on quite a lot of this in terms of what the pros are of biocontrol. Obviously, it's environmentally friendly. You're not spraying stuff all over the place. It's not polluting the environment. There are low risk of non-target effects. And as I said earlier, we've had no adverse non-target effects in South Africa in the history of biocontrol. So we have a good track record. It's on the whole self-sustaining. I mean, in some cases you do need to do these inundated releases where, as I spoke about with water weeds, some of the other weeds where you need to release more than once. It's cost effective. I mean, I know chemical control is exceedingly expensive. Mechanical control can be exceedingly expensive. And I think if you look at it over, if you compare the three forms of control, it is a cost-effective form of control. Generally, the agents will disperse themselves. I mean, we've seen quite a few of these agents that I've discussed don't need implementing. They're doing their own thing. And it's also permanent. There's not really any reversing it. The weeds can't run away from natural enemies. Weeds are in one place. It's that natural enemies that are moving around. And then if we look at the cons. So obviously, again, I touched on this earlier. It's fairly unpredictable. And, you know, we always ask as biocontrol scientists, well, you need to predict how successful this agent is going to be. It's very difficult to make those predictions. Much as you always have a gut feeling about, you know, this agent looks really good, it's doing, some agents that you see in their native country actually not particularly damaging at all, because there are all sorts of other things, parasitoids and whatnot, that are actually suppressing them. But we just get a gut feel of something that's going to work well, but we're not always right. So success isn't guaranteed. And as I've mentioned, there are considerable efforts involved in getting a new agent. I mean, I've been through the whole process. I need to elaborate on it. It's a lengthy, time-consuming process. And also, sadly, I think it is never really integrated properly. And this is a really important factor here. It's never integrated properly with other control methods. You know, we usually say biocontrol alone is not going to have the sort of impact that people think it's going to have. It's really important to, you know, it's fine. You can use chemical control, you can use mechanical control, you can integrate them, you can integrate all of them. And that's actually how these tools need to be seen as being integrated. And obviously, again, it's not one recipe that's going to fit everything. But there are ways of integrating the control methods, and it's really important to do that. I think another thing, and again, I touched on it earlier, it's expectations. public expectations are extremely high and often too high. You know, you'll supply people with biocontrol agents, and then they come back to you and say, well, you know, they haven't done anything. My trees are still there. And you think, well, I did explain to you that it's not going to affect your trees. The trees will always be there, but it's reducing the seed, or it's reducing the flowering, but people listen to what they want to hear. So, and I think they immediately think, you know, chemical control is costing me a lot. Let's get biocontrol. That's gonna resolve my problem. And that's part of the problem, the expectations. And you can try and tell people, but they don't listen. So that's one of the cons. It's something that we are constantly having to reiterate. But, and I'll make the point there, that it is a particularly slow process. You should never really see biocontrol as being a quick fix, because it isn't. It's there for the long term, and think of it as being something that can assist you in the long term and slow things down in the long term, because it does work in the long term. I think that is, I don't know, okay, so just a few important points to remember, and we kind of like broached on this a little bit earlier, and that is that timing is really important for the collection of your agents. And sometimes what I usually do if I'm collecting and going to be sending off, I put something, twig, put a pod, paper toweling. The paper toweling is critical for all the reasons that we've spoken about. Don't keep too many insects. Again, we spoke about it. Don't keep too many insects in the containers. You're going to get death. Keep the insects cool at all times, and that is really critical. And as Willem said too, you know, you don't want to be leaving your cooler box in the back seat of the car in the sun, or standing in the sun next. Put it in the shade always. Keep everything, and it is really important. Keep emptying your, you know, whilst you're sucking up your insects into your fruiter, keep emptying them into jars. Don't suck up till you've got 10,000 in your fruiter and then separate them. Just do it. Make sure all the time these things are being kept cool. And then obviously release those insects as soon as possible. Don't hang on. Much as we know, there are ways of trying to keep them a little bit longer, and some are easier to keep a little bit longer. My suggestion to anybody is, once you get your insects for release them. Release them immediately if you can. And that's, I think, it. That's really all I have to say.
I'd just like to speak about your previous slide where you talk about integrating different methods, control methods. So what we did in Worcester, we're trying it now also in Lagos, but we used to live in Worcester. We released the fungus on Hackia in a very high area, so it's like about a 3 to 4 hour walk to the area. So you don't really want to send the team in there because it takes a lot of time just to walk into the site. And it was also like really dense, so it takes a lot of time to cut it down. So what we did was we released the biocontrol agent, and then we kind of forgot about it, worked on more accessible areas, you know, easy to get to, lower density. And then after about 2 years, we thought, okay, let's go and see what's happened up there. And yay, it was particularly wet season, so the fungus did really well. So instead of having to clear, I don't know how many trees per square meter, you know, it was now a matter of how many trees per hectare. You know, so instead of spending 40 person days or a week or something, we could actually reduce that. So it took a week to clear the area, whereas before it would have taken us 40 weeks. So to integrate those two methods really does work, but you need to kind of choose. You need to be site specific, look at the pros of that area of releasing that biocontrol agent, whichever one you're going to use. We work with fungi because I know fungi, and you know, I mean, even in some of those trees, you could see there was evidence of insects. So you know they are there, but yeah. So I firmly support that. Definitely integrate control methods. I think Ellen said it before. Biocontrol isn't a silver bullet. You know, it's part of a package. But you can definitely use it to your advantage.
Yeah, I mean, we've certainly seen with workarounds, and look, not everybody is able to or wants to use fire as a method for control, but fire is actually a very good way of controlling in an integrated manner, controlling oikons, because you're really going to get on top of your seed banks. You know, and we've seen it. I've worked a lot in that. It's next to work in the Denell test range area. And it's quite amazing. After 2 fire cycles through a big patch of roicrons, there's now, there's native vegetation coming back. And that's thanks to having both fire and biocontrol in place. So, you know, I think the problem is we often look at weeds. You know, yesterday we were walking around here and I just thought, it makes me feel quite depressed when you start looking and you think it is such an insurmountable problem. So always then try and focus on what can be achieved rather than what can't be achieved, and focus on the winners. But certainly over my years working with acacias, I now really do feel we can get on top of roicrons. We really can. It's one of those that is far easier to control than any of the other acacias. It's a winner in terms of management, and it's a winner in terms of integrated management. So
yeah, there's a Jongersfontein, a guy who inherited a piece of land with a house from his uncle, and he retired there now. It's about 100 acres, but it was all overgrown oikons. So he decided he man alone himself, 4 hours a day, 5 days a week. He's going to remove oikons. 18 months. He cleared 100 hectares. Then he started the property next door on top at the Homfontein is municipal land, where the sewage dams are, but it's quite a popular cycling and hiking area. He started clearing there, and some of the people who came there started joining him. And they had a team now, and they started doing this, and they started talking and so on. That area is now a declared nature reserve. It happened last year. And then the neighbor on the other side has got these dunes going down to the beach. It's just one big workaround spot. He's cleared a third of the dunes now. And it's just one guy who started all by himself, consistently doing it. And I think that the big thing is when you deal with landowners also is consistency. And that they, in terms of management, that plan every year to do something and not think that it's a once-off.
I think too, one of the differences between, say, roicrons and black wattle, the seed doesn't live so long in the soil. We're pretty aware of that. And it is more susceptible to fire in terms of seed loss. The others seem to, it causes, and sure, a fire will cause germination, but a lot of the seed gets lost. Whereas with something like black wattle, you've just got a lot of seed, it loves fire, it regenerates vigorously after fire, and it lives for much longer. We've got some seed burial trials actually in the Rook Nature Reserve, and it's very interesting. We've been pulling out these little containers that have had seed buried in them for up to 20 odd years, and the roicrons actually disappears quite quickly. Yeah, so there's always that portion of the seed that it's tough. It'll hang around, but it's a relatively small portion. Whereas with the others, Port Jackson, black wattle, that seed just stays there, and it's alarming.
Do you have
So there was a question from Jessie there.
For the seed bank, for the seed burials, we actually haven't published it yet because we've still got, and I'll probably be on a Zimmer frame by the time we do the next, or I'll have to tell someone where the seed's been buried.
Even
dig
if
it up.
I can just see the
Yes, I can show you.
Because everything I tell people at this point is, well, not everything is secondhand. Sorry.
Sorry, we just had some questions here, Commissioner, and then Jessie as well.
It's just to add in with what the gentleman is saying over there. So I know there's Dr. Dion Biergeman, he's got a hacking team there. It's just unfortunate that not even the municipality is even there. Because most of the hacking is taking place on municipal property in Stilbaai itself. And they actually, they are benefiting with the fact that knowing there's a hacking team, basically people from the community. And for them to actually join with them and doing it would have been great, but that's
Yeah,
just
so
sad.
I mean, so I work with them quite closely. So, yeah.
But Jesse, you had enough for.
Yeah, sorry, I have like 3 things to say. With that on the Roy Krans, Creekstoel was like a developed area and they had to bulldoze like the sand dunes and rebuild them. So there was nothing, it was just Roy Krans, and then they took everything out. And that whole area is just indigenous and there's like a tiny patch of Roy Krans. So I think it's like you said, like keep going, talk to the landowners, keep doing the alien clearing. Which I think is cool. I wasn't involved, I just observed it from afar. Second thing is, I wanted to know what is the rules and regulations of releasing biocontrol in your plantations? Because I don't know them.
So,
because most of those plantations are connected to pristine like nature reserves or like private land. And those people want to release it. Yeah.
This is being recorded, isn't it? So, but you're talking about, so the plantations that they're only worried about are not in this province. We're talking about plantations up in KZN, the main growing areas for black wattle. So KZN and Mpumalanga. So it's a certain area south of the N4 and parts of KZN. It well, it's not, I mean, legally speaking, there's no law that says you can't. We just had a gentleman's agreement with the wattle industry. So they, and this stems from years back, where they said, okay, we will give you permission to release the seed feeding. We will give you permission to release the midge, for example, but please just avoid, I think it was actually 100 kilometers from our plantations. And, you know, if it's going to be, they actually said, if it's going to be in this area, just let us know. Let us know where you intend. And actually, every time I did any release that happened to fall in that area, well, they were quite happy. They gave the go-ahead. And I did say to them that, look, obviously, in time, if this insect does establish in these areas, it's going to spread. Sadly, we didn't have establishment. I mean, it's established in very few areas in Natal, mainly where the sort of misspelt region. So Nottingham Road, Dog, all those sort of areas where they get quite a lot of moisture during the winter months. They don't get rain necessarily, but they get moisture. And the same in Mpumalanga, we've had a little bit of establishment. So it hasn't, I think it hasn't really raised any alarm bells with, but I think if you wanted to, nowadays, I mean, and there's also nothing stopping people from grabbing, as people do, they will grab galls, but they don't always know what time of the year is a good time to collect them, but you know. So nothing stopping you from doing it.
So the argument there will be how much of their wattle has spilled over onto your natural land.
Yeah.
Your biocontrol agents spill over into their land.
Yeah. Well, they always claim that their wattle plantations are so well managed, and actually the jungle wattle's that's not their problem, because it's not their plantations are managed. So
yeah, just to come in with that, yeah. So for us, especially on reserves, so the development you're talking about is adjacent to Biocontrol Nature Reserve. So that it's like you said, Fiona, it's good to inform the reserve manager as well, because if it's going to be continuously monitored, that the reserve is aware about what's going on, and especially when it comes to the spread of the biocontrol on the reserve, because I'll take the also, for instance, if there was biocontrol to be released adjacent from the reserve, we'd like to know about it, because if it comes in, it's going to be a nice thing for us as well. So it's just good to know about that. But that collective development is something else, because that influences dune movement as well.
Yeah. But what are the plantations? Plantations of
Roy Krans.
Roy
That was
Khan.
not plant, I mean.
No,
that
I
is
just
the-
asked in
Oh,
general.
you were just in
Yeah,
general.
we don't
asking.
have
Oh,
like black wattle plantations here.
No. I mean, I know there are some up in the sort of, further into the Southern Cape, Uniondale area, there was some plant, but whether I think they're sort of old defunct plantations now, of both green and black wattle. But, yeah.
Other quick questions? Yeah,
Um,